So our first speaker, where I put my glasses? Let's invite our first speaker, Carla Carvalho, and she's going to present biomonitoring fossil fuel contamination on an anthropic coastal estuary, the Radical project. I think it's another one, not that one. Little technical problem. Okay.
Yeah. Thank you. Good morning, everyone. Thank you for being here after the great night we had yesterday. We have dancing and drinking, so thank you very much for the support. And so I will talk about this radical project. This is the first time that I talk about it. This is a project that involves me and three former students from the university that I work. It's Fluminense Federal University. The first one is Bruno Libardoni, who has a family, an Italian family also. And he was a former student for another professor. And one day, we are just at this, crossing this view, this beautiful view from Rio de Janeiro, which is just besides our university, our institute. And we are talking about measurements of biogenic fraction and also a project that he just won a few days later, before, about this. I don't know if everybody can see here, Caravella project. It was a project made by Bruno Libardoni and FURF Design. It was an eco-design project that won a prize in China in 2018. So this project have some algae on the bottom of this kind of boat and is made for cleaning waters. And we are talking about cleaning waters or measure the oil contamination in waters. Because here, as you see in this beautiful view, we have the Guanabara Bay. And the Guanabara Bay is a huge place that receives lots of contamination from every place. So there are lots of people just living around the Guanabara Bay. There are some factories. There are some many boats that cross this bay daily, and so it suffers a lot. So talking about the water pollution is the largest source of contamination in the aquatic environment. So besides the nutrients that can make changes in the environment, the excess of nutrients, the main estuaries are rich of persistent organic contaminants, and also they came through rivers and also due to the oil spill and another kind of contamination in the surrounding environment. So like this one, it's not a good view to see. There is this oil spill in the northeast of Brazil in 2019. It was really complicated to deal with it at the time. It was crude oil in this beautiful environment that attracts thousands and thousands of people to enjoy the beaches, enjoy this paradise place. So what is important is if there are large spills, large oil spills, it's easy to see, but it's a little bit difficult to deal with it. But the main problem is when it's kind of invisible. Small spills presents a chronic problem and could cause a potentially cumulative effects. So as you see here, it's also a view from Guanabara Bay, almost the same view from the first one. And we can see here that we have this platform. So since we have places to fix some ships and platforms around the bay also, they are used to stay there for a long time. And we have this kind of problem, just like this one. Can you see the big quantity of oil that came from this platform? So, in fact, this oil can go through the water column and can suffer many kinds of mobilization. So we can have this kind of dissolution we see on the left side and goes directly to the remobilization on the floor, on the sea floor, and also biodegradation and in different deeps of the water column. So the question is not so complicated. If we are dealing with algae, can algae be used to monitor and remove pollutants from the water? So since algae is the large photosynthetic organisms that are able to assimilate these nutrients and then can fix the CO2 also and can be transform these contaminants in huge amounts of biomass. So if we can measure the oil content in this algae and use it just like not only a biomonitor but also a bioremediator, it will be awesome. So, in fact, there are lots of algal turf scrubbers. There are this kind of system that is already used to retain the contaminants from water, and such as metals and pesticides and other kinds of contaminants. And thinking about this, we decided to try to make a prototype of the Caravella project, using cheap material, and it was a PhD of my student Daniel Tremel, who is the third person in this co-authoring work. So the algae growing process on every kind of substrate is just like that. So first we have this bacterial conditioning for the substrate to be grown, this EPS, and also after that we have the colonization matrix by algae, different kinds of algae. We cannot choose them because they are in a natural environment. And the growth and reproduction of these algae cells starts to become important. And some algal and bacteria symbiosis also appears. And after that, we have a mature biofilm matrix that is densely populated with these algal cells. So if you search for bioremediation and algae in some studies, searching for titles and abstracts, that is the cloud that we found. Many metals, heavy metals, are measured and detected and removed from water, but the oil content in this papers is, there is a few works about oil and bioremediation. So this is why we plan to do this project that is Radiocarbon and algae radical project. So we have 3 phases. At the first one, we did a validation in the indoor experiment in the lab, just picking some samples and measuring the PMC of the sample in the natural environment just to check if they were in equilibrium with the atmosphere, and then use them as natural material for contamination, intentional contamination in a controlled environment. And the phase 2 is the construction of the prototype and put it to work in the field. So it's an outdoor experiment. It was made during the pandemic time, just in the end of it, so it was difficult to deal with it, but we could measure, and I will show you the preliminary results. And also in the phase 3, that is the one that's ongoing now, there is a discussion of what we can do with the biomass that we recover from these structures. It's contaminated by oil, by metals, by which kind of materials, and what we can do with this biomass we recover. So this work is related with the SDG, basically, with the SDG 14, that is a life below water. For us, it's very important in this environment. But as we will see after, there are many others sustainable development goals that we can achieve with this project. So our place of study is the Guanabara Bay, as I told you before. So it's a rich environment with a biodiversity. There's life there, but it suffers a lot with the surrounding people that lives there, the surrounding contaminated actions that happens there. And so it's interesting to understand a little bit more about the contamination in this environment. So here we are, this is the Guanabara Bay, as you can see here on the bottom. I will show a kind of a dynamic movement of the water getting in and getting out as a breathing system during one day. So I will show you just like that. So this is this dynamical study is from another project realized in Brazil from the Rio de Janeiro Federal University, and you can access this kind of data in this QR code if you want to. So as you see, it's a dynamic system, but it has lots of surrounding cities, and also in here, I don't know how to put this on here. As you can see here, there are mangroves. That's the link with my colleague presentation during the Wednesday session. So there are lots of mangroves in here that has also a biodiversity, a big biodiversity and a life going on. And so the phase 1, the microcosmos controlled experiment happens in our lab, not the radiocarbon lab, but the other one that I also run. It's in the chemistry department. And this is a paper that is on radiocarbon. It's the work of Titolo, who is the fourth co-author. It is in here. As you can see, were pandemic times. And so what we did was we received some fuels from the ANP, the National Agency of Petroleum, and we measured the content of this growing contamination in the algae using kerosene and also diesel. And what we found, it was a kind of growing concentration, not too much easy to see for kerosene because it's very volatile. And so we measure the differences between these concentrations we have. So we validated the technique that we can measure by measuring the radiocarbon fraction in this algae system, the contamination in the environment. So we move forward for the phase 2. There was this PhD of another student, as I told you. So the idea was to make this simple use this simple strategy with cheap material just to make this floating structure to be colonized by this algae. So this is the kind of croquis, and this was an experiment during also to find temporal variability in the time. So we divided in 4 pieces and collected 1/4 at a time. And we installed 3 different structures in 3 places that we assumed to be more or less contaminated. So as you see here, bar is the top one, means in Portuguese barcas, that means ferry boat, and BV means Boa Viagem, is the name of the beach, Boa Viagem Beach, and mar means mariculture. It's a farm for growing seafood. So this is the first one that we made. It's just nearby this ferry boat station. Thank you. Oh, it's 5 here. Okay. And there's nearby this ferry boat station, and you can see the algae growing on it. And the second one is BV. Some of you have seen this museum before. It's the Contemporary Museum of Niterói. And this is the mariculture farm. There are lots of growing sea shells in there. And so just for you to note, in 19 days we have this amount of algae, so it grows fast. And we recover this material. And this is only the first slot of collecting. We have some pictures of it taken by drone. And we have 2 campaigns, one in 22 and one in 23. And so these are the main material that the main techniques that we use it, we analyze it. And as you can see, there's life there. And so we divided the one part of each recovery material into 9 pieces to have this statistical data. So then we have these protocols to analyze also water samples, not with radiocarbon, but the geochemical markers. And also the algae for all of this analysis, but for C14 we used this method. So these are the preliminary results. So as you can see in the picture of the left, it's possible to see the oil in the surface water. So this is the reason why we planned to make this structure floating, because it will be able to retain this oil before it comes into the water column, degraded and incorporated into the system and into the ecosystem of the living animals. And also ours, because there are fish there. And so we did the N-alkanes just to try to check what kind of oil could be present, but actually we have this, it's not a signal just like in that other paper, we have a different kind of bimodal signals that could be different kind of oils. So it's difficult to know which kind of oil is that, but it's kind of a mixture. So this is interesting. We did also the metals analysis, and we have this correlation with these metals that are related with the petroleum extraction. And we also look in detail to the algae, and we discovered that these diatoms that are present there are seriously degraded also. And what about the plastic? It's everywhere, right? So we have lots of plastics that were retained in the structures also. So you can also use this for removing the plastic in the surface before it turns into smaller pieces. So this is some results we had using the FTER. So we had this 97% of microplastic retained in one structure after 4 weeks. So I don't know what you think about, but I think it's huge because it's too much plastic. And now, and also we have there a project that was made by a student from another laboratory, this one that used to help us with the FTER. And she found lots of microplastics in mussels from some fishes from Guanabara Bay. So if we can retain them before they get into the fish's bodies, it will be better. So this is just to show you the biomass production during this time. There is some ups and downs because it depends on the sunlight, depends on the weather, depends on the movement of the sea. So it's a little bit complicated, but there's lots of biomass in a short time. So I return to this question. Can algae be used to monitor and or remove pollutants from water? Yes, it can. It's possible to use the algae. As you know, there are lots of papers doing this. But we use this kind of station in this project to measure and to remove many kinds of contaminants at the same time. So this project works with biomonitoring, multi-process analysis, and also bioremediation of plastic and microplastic. And there is the biomass production, what's interesting. Also the biomining, because if you recover metals in the future, maybe someone can use and recover these metals. There's lots of studies on this subject. And also biofuels. So this is the third phase, as I told you. And we have this biogas studies going on, the pyrolysis, and also another way to make briquettes. I don't know the same in English, I'm sorry. To make some other kind of fuels. And also education, because this is an educational project. We go to the schools and show this and bring the students to the lab to see what's going on, what kind of pollutants we can remove using a nature-based solution. So it's also an action on the UN Ocean Decade that is almost in the middle. And we have this, many of these SDGs achievements, at least 12 of them. So I'm finishing my presentation, and I would like to thank a lot the laboratories that work with us, because it's not, we cannot work by ourselves when we're doing this kind of project with lots of measurements combined. And also the founding agencies, the Brazilian founding agencies that give us money to do this. And these are these two projects that are coordinated by me, one from Scientifico and another one from FABIO, that is another institution in Brazil. And also, I would like to thank the Niterói mayor and the ferry boat company that make it possible to deal with it, and the Maricultures Association that helped us to maintain the structures there. So this is my takeaway message, and I would like to thank you all for being here, and this is my email if you want to talk a little bit more.
Thank you, Carlo. We have time for a very quick question. Okay.
Thanks for this very interesting talk. What is the overall aim of your project? Would you like to put that into action in order to produce floats at the bay, to have them continuously collecting pollutants? And what will you do with this material? Or do you have an idea what to do with this material later on?
Yes, as I told you, it's a collaborative project. And this thing that I showed you before, the Caravella, it's a project from Bruno Libardoni. And he has this enterprise to put these structures around places that need to be cleaned. So this was a kind of joint project with former students at the university, and the university helps the students to go on with this project. So he has the enterprise, he will get the money, and I will do the analysis of radiocarbon. So this is one thing that's going on now.
Collected this material, what to do with it? I mean,
I take to the lab.
Press it, dump it, I mean...
freeze dry it. There are lots of freezers with materials because we cannot measure it all for now, but we keep going. So we have some with this professor from Rio de Janeiro Federal University to trying to extract biogases, and pyrolysis one. There are lots of labs involved. So I am coordinating these labs to make this work more and more, and to try to extract the biogas and other kind of recycled fuel that will be possible to use after.
Okay, let's thank you, Carl, again. Now we are going to have a talk from Kita Macario, Itaipu region in Niterói, Brazil. Cultural and natural heritage under threat from land speculation.
So good morning. Thank you for having us here, all of us from Brazil in our tropical environment session. And I'll show you a little bit about an ongoing project which has this multiple purpose. And one of these is to save a specific place in Itaipu, which is a place in Niterói, in Brazil, the southeast of Brazil. And I'll show you some of the topics of my presentation. And then we start with biodiversity in Brazil, so why Brazil is important in this context. So Brazil is the #1 in biodiversity, taking into account all different kinds of biodiversity. So it's a very important place for the world, and we have very varied environments and biomes. We have the Amazon rainforest and Atlantic forest, and also the Cerrado, with a lot of different ecosystems, terrestrial and aquatic ecosystems. So this is just to make you feel bad about the animals, but then we have a lot of different kind of species, and one part of what we aim always is to preserve and conserve our natural heritage. So here we can have a look in at Guanabara Bay, as you saw in the first presentation, and we have a zoom in the Itaipu region. There is this place, the beach is called Itaipu, and the other one is Cambuinhas. And this is part of a place where we have this mountain, which is Morro das Andorinhas. I don't know if I can use the pointer here. And it's a protected area. And we have not only a lot of biodiversity, but we have a lot of shell mounds, so archaeological settlements. Itaipu is how it's called this place. It means, in Tupi language, it means the rock that sinks. So it's been this area, the southeastern coast of Brazil has been occupied since about 8,000 years before present, but mostly about up to 5. This is what we have. Most of the remains that we find are up to 5,000 years, and but there's only one date from a long time ago, made by Professor Lina Kneipp, which was an important archaeologist that used to study the shell mounds, and it was dated like 8,000 years before present in this specific place. But we don't have a very good record, and most of the shell mounds in this place are destroyed or apparently destroyed. And this is an important place also because we know that Darwin visited Brazil in his first expedition, and this was the place where he first entered the Atlantic forest, and he was amazed by its biodiversity. Here you can see in yellow, how can I use the mouse? Over here, it's from the Diary of the Beagle, where he talks about Niterói. So Niterói was the first place where he entered the Atlantic forest. And because of this, and for this recognition of biodiversity, it's a place that is considered by and was recognized by UNESCO as a World Heritage. And then today there is this pathway that they call Darwin Pathway, where you can walk and know the place, and it's a place for conservation. So this place specifically, because of the shell mounds, because of the biodiversity, it's a place where we do a lot of studies using radiocarbon. So most of what we've been doing, it's looking at different shell mounds on the southeast of Rio de Janeiro. So we have in these different colors with the letters, some places that represent some shell mounds. And then this picture is from the newest one that we analyzed. It was published last year. The Duna Grande shell mound, which is not really a shell mound. There's this complication between archaeologists, but we cannot use some specific word for this before we are pretty sure it's the same kind of mound. But it was dated by us. So we usually tackle this marine reservoir effect using different approaches. So one of these approaches is to use the samples from expeditions. So they are all over the world in many different museums. And my former student Eduardo Alves did this when he was in Oxford for his PhD. And we analyzed a lot of different samples from different 19th century expeditions. And we looked at the marine reservoir effect all over the Brazilian coast using this pre-bomb analysis. But the main subject that we like to study, to see the past environment, are the shell mounds. So the shell mounds are places where you can find a lot of cultural heritage, like cultural remains, but they are naturally places that you can also find remains that represent the past environment. So this, like here, you have like otoliths, and these are shark teeth and barnacles, and these are marine and terrestrial mollusks that we use for studying marine reservoir effect. Oh, I forgot to make the point move. So the way we do it is having not a pair, but a lot of different samples from the same archaeological context. We always work together with archaeologists. There are experts in this kind of settlement, so it's not we go there and collect samples for ourselves, but we have many different collaborations. And the approach we use is usually using Oxcal. So we have this phase where we analyze samples from marine and terrestrial origin, and we come up with the distribution of the delta R. And the last work that we did about it was always adding some places and shell mounds from different time ranges, because not only we wanted to see how they were, the behavior like in the context of the marine reservoir effect, but also during time. So we wanted to look at the spatial differences, but also the time differences. And then just to make sure you understand, I'll come back so I make sure you understand. Each of the point, it's not a pair of dates, and not only for one big shell mound, but a specific context from a specific time with between at least 6 to 30 samples for each point. So we have these distributions. But then here it's plotted as the median. And so we have the one standard deviation, and we could see change during time of the marine reservoir effect based on these mounds. And especially the one in yellow, which is the Duna Grande site, that this is the one that we last worked on. This place is, specifically this site is protected by the federal law, and so it's safe for now. And we also work with a paleo sea level reconstruction. So for local sea level, we usually use vermitites. So we have some works on this too, and we have this reconstruction about how was the sea level, the relative sea level in local scale at this area. So we got a lot of flooded environment, which is very characteristic of shell mounds. But then if we put this together, we can see a pattern. While we have the sea level decreasing from 5,000 years ago to now, we have an increase in delta R. So this is something that we found out with the first mounds, but then after we got this older Duna Grande mound, we could see that we had this kind of pattern. And so we are aiming to study the oldest mounds, and we are trying to find them. So I want to talk about cultural and natural heritage under threat, because this is a place where, of course, I think I just proved to you that it's a very important place from the biodiversity point of view, but also from the archaeological point of view, because these are the most antique dates that we have for this kind of culture, and they are in this specific place. And so we know that because of climatic changes, we've been having different threats all over the world. So it's been reported in many different papers and many different areas. We know that the sea level may increase, and we have to run to take the samples or to study the environment and do what we can to protect because of climatic changes. And of course, there are scenarios that are estimated for the next decades, and this is something that we have to think about. But we still have some time. But what we have in this place is not only the threat by climatic changes, but I told you in the beginning it's an environmental protection area, but it's not the whole place. So if you look at this map, you can see that the part in green are the limits of the protected area. So the exact place of the archaeological sites, those that are the most ancient that we know that exist and that were dated before, it's not protected. And in fact, there's been a law last year, the municipal law, that enables the land use of this place. So it's really, we have to hurry to protect it. So this is for you to have an idea what kind of crazy things this kind of people think of doing. So it's a project of having fake islands to sell to very rich people. So they wanted to destroy the whole area and build these islands. And this is not over. We still have time to protect it. But one of our aims is to study this very fast so we can find the oldest sites and, make use of the news so we can make the world see this place and help us to protect this. And of course, there is the environmental and the cultural part. So there's the Museum of Archaeology in Itaipu, where they have a very important work with the community. So they show the expositions, they talk about the importance of cultural and natural heritage. They have many different projects in education. Also, they have a very good relation with the fishery village, because they still have the artisanal fishery like for more than hundreds of years, and they also are in the middle of the way of the construction sites. They want to get rid of the fisherman village. And so this community is very active. As you can see, they made this SOS image with themselves, and this is in this website where they call it a lagoon forever. And just to show some of what we are doing now, I just got this date like this week, so I wanted to add this. In this area that is not protected, especially the place that we know that where the oldest dates are, but the population believes the sites are already destroyed, but you cannot see the like shell mounds like the other places. It's just buried. So we made these test pits so we could see and try to date different materials, mostly shells, so we could look for the approximately time of the occupation. Of course, this is not an archaeological digging. It's different. It's just an initial approach to find the shell mounds. So we found that the oldest dates is where you can see the star, and so we begin to have authorization to dig in this specific place. So my conclusions are that archaeological and marine remains on the coast of Rio de Janeiro have demonstrated temporal variability of carbon-14 marine reservoir effect. Of course, this is a place I've not shown you in detail, but it is a place where we have competing fresh water and upwelling, because this is an important upwelling area. And it's difficult to separate what is the time variation and what is the different kind of environments. And we see this pattern of increasing delta R as a relative sea level decreases. And what we are doing now is to map the area under threat to avoid the destruction of the archaeological sites. So we want to make everybody know that the sites are already there, and then they're not destroyed, so they can still be saved. And we believe that finding this area will contribute to this, and also to our study about the time before the sea level started to decrease. So we want to know if regression and transgression, of course, we believe this will have different effects in the delta R variability. So those are my references. And I'd like to thank all involved people, especially the Museum of Archaeology from Itaipu, and my colleagues, and also the ones that are in this new phase of the project, especially those who helped us to prepare the samples in a rush, and the funding agencies. And I'd like to, it's not good shape, the writing part, but I think this is important to say, since we are in a radiocarbon and environment, and I'm talking about a little bit about archaeology also, but the World Heritage Emblem that I showed before, it represents the interdependence of the world's natural and cultural diversity. So I think it's a thing that we have to think about, not to separate science. And so I'm going to sing. Okay. Just breathe. I've been practicing my Italian, so penso che un sogno così non ritorni mai più. Mi dipingevo le mani la facchia di blu, poi d'improvviso venivo dal vento rapito e incominciavo a volare nel cielo infinito. Now you can volare, oh, cantare, oh, nel blu dipinto di blu, felice di stare lassù. Grazie mille.
Thank you, Kita. We have time for a couple of questions. Any questions for Kita? One?
Peter, thanks for the talk. I just have a question about delta R and the sea level. And you mentioned about the competition between the upwelling and the fresh water.
Yes.
Supposedly, when you have a lower sea level, that means you have more fresh water contributed to the side. Is it what you mean, right?
Yeah, I think the problem is not having lower or higher sea level, but to think about transgression and regression, because it's a different framework where we have the sea level increasing or if it's decreasing. Because this area is an area of mangroves, so it used to be flooded by the time of like 5,000 years. And since the sea level has been decreasing, so the mangroves just accompany, the coastline accompanies the decrease in sea level. And the sites are in the same place, but the places where they find the mollusks is going away. So this is kind of a complicated thing to think about the variation in time, but also we have to think that this area has important upwelling. It's not a very pronounced, but we thought that like the shell mounds that are closer to one specific area where you have upwelling, where you can have the upwelling signal, and there are other works with foraminifera that show that this influence has been going on for thousands of years, but we are not sure how much the upwelling reaches this place and how it can influence. So it's kind of complicated to have both things at the same time.
Okay. Have you ever measured any shell in the river to see currently where the carbon-14 in the fresh water, because it depends on the geology. They can maybe old, right?
Yeah, we don't
have you measured anything there and see?
Yeah, we don't have in this area any influence of all the carbonates. It's an area that this is not something that happens. The geology of this area. Okay, so we also have delta C13 measurement, and we can separate, especially when we did the 19th century expedition, we did the delta C13 for all the samples, so we can separate which ones they belong to marine and freshwater environment. So this is kind of easy to see this difference, but how much it can influence each shell mound and each source of shells, where they get the shells from, it's not so easy to understand. So this is a result that we still have to understand better, but I think it makes sense.
Let's thank you, Kita, again.
Thank you.
Now we are going to have the talk of Ingrid Shankar. She's going to tell us about how long does carbon stay near pristine central Amazon forest, an empirical estimate with radiocarbon.
Hello, everyone. Yeah, I hope you can hear me well. So, yeah, good morning. We move now from the water to the terrestrial part, but we keep in Brazil, okay? So, yeah, this work I started during my PhD, and I further developed it in my first postdoc, and it was submitted to Biogeoscience Journal, and through that QR code up there, you can see it, you can see the ongoing discussion, and it's under review now. Okay. So, yeah, I would like to start saying that this work is also in memory of Ingeborg Levin. She was part of my PhD advisory committee. And this is a picture that took in 2020 when we are discussing the very first results of this work, and she made this very nice scheme, and we are discussing the next steps, and yeah, that brings really good memories. Okay, so yeah, tropical forests, as you know, are very important environments. They assimilate a lot of carbon through photosynthesis, and especially the Amazon, as is the largest continuous tropical forest in the globe, assimilates about 16% of the total terrestrial gross primary production. And here it's a global map where you can see the red. Sorry, I want to put a pointer. I always struggle with that. Let's see. Yes, okay. So yeah, you can see the red parts is where we have the largest DPP, and here the Amazon is completely red. But one thing that we have to keep in mind is that it's important to know not only how much carbon a terrestrial ecosystem can store, but also for how long. And that's going to be my aim here, to discuss this time aspect. And to do that, I'm going to use this concept of transit time that maybe you're more familiar with residence time or turnover time concept. Transit time, it's, I would say, broader in the sense that it explicitly considers the different time scales of different carbon pools in terrestrial ecosystems. So carbon, here in the figure you see carbon enters through photosynthesis, but because in different carbon pools, they're going to have different turnover times due to the carbon dynamics of each of these reservoirs. In the final respired CO2, we have actually a mix of ages of carbon. So then we can discuss this total ecosystem respiration age as a probability density distribution of these different times. So, okay, so then the idea here is to taking from this large photosynthesis that entered the Amazon and to think how long does carbon stay in a near pristine Amazon forest, and for that we're going to aim to calculate a mean transit time in principle. And there are other works dealing with this time concept, trying to estimate this time that carbon stays in tropical environments. First approach that I'm showing here is of the turnover time, which is simply the stock over flux. So the total stock, in this case of soils, over the photosynthesis flux from that map. And in this work of Cavalhais and co-authors in 2014, they estimate that tropical forests have a mean turnover time of 14 years. Yeah, but spanning from 1 to 2 decades. Another approach is to model the whole distribution of transit time, and to do that, to use carbon pool models. And in this case, it was for a tropical forest in Colombia, about 2,000 kilometers far away from my study site. And the nice thing about estimating the distributions is that we can get much more information than just the mean value. So for example, half of the GPP in this case takes less than a year to be respired back to the atmosphere, but if we consider the 95% interval, it can take up to 70 years. Nevertheless, the mean transit time is also around 1 decade. And finally, a third approach that I want to show here, because I'm going to later compare to my empirical estimate, is this one, made by Susan Trombor and De Camargo in 2009, where basically they estimate the turnover time of different compartments in a forest plot close to Manaus, in the middle of the Brazilian Amazon. And the total ecosystem respiration they estimate is between 3 and 7 years. So we were thinking about how we could actually get an empirical estimate of the mean transit time, and we thought, okay, why we don't simply try to date this ecosystem respiration. This has been done, to our knowledge, only once before in a temperate forest through the approach that I'm going to show you. And basically, okay, with that, we come to the questions. What would be the mean transit time of carbon for Amazon terrafirm forest estimated with killing and milliturns methods using the 14 CO2 from ecosystem respiration? And how does this empirical estimate compare with other model-based estimates as the ones that I showed before for the mean value? Just a quick revise of or review from the killing plot and militants approach. So basically the killing plot, it was largely used for Delta 13C. Of course, sometimes, as I mentioned before, it was used for the Delta 14C too. And the main idea is that through a regression between the isotopic signature and the inverse of the CO2 concentration, in the ambient air, you're going to have the signature of the main source. And the millitants model, it's actually very similar in the way that they use the same basic equations, or the balance equations, but the difference is that there is a rearrangement that you don't need the background to be constant anymore, so you can incorporate the information of backgrounds that change over time, and that's also very nice for us because we expect a variability on the 14C in our background here. And the only difference to interpret these regressions is that in the case of the killing plot approach, we're going to look into the intercept to have the isotopic signature of ecosystem respiration, and in the militants regression, we look into the slope. Okay. So, so here's just to give you a view of the ATO site where I collected my sample. So ATO stands for Amazon Tall Tower Observatory, and it's this beautiful tower, 325 meters in the middle of the Amazon. And here, just for comparison, the Eiffel Tower, that when I started working in ATO was a bit larger, but I think it was 2022. They added an antenna, a TV antenna in the Eiffel Tower, and now the Eiffel Tower is a bit taller. Well, what we can do. But still, ATO is the largest structure in South America. And just to give you an idea of the dimensions here, the canopy level is about 35 meters high, so it's comparable to the height of the Cristo the Redeemer in Rio de Janeiro. And we have also two other towers, 8-meter height, that are comparable to I always forget the name of this, but you know, this other structure here. Okay, so how was my sampling design? So basically, I use one of the 8-meter towers to collect sub-canopy level samples. So at 4 and 24 meters, I collected through a whole diurnal cycle in October 2019 and December 2021. And I also collected, so in 2019, I also collected 79 meters to work as a background because we didn't have still measurements for 14C from the top level, sorry, for CO2 concentrations for the top level of the tall tower. So to use as a background for the millitons approach, I used the 79 meter in 2019. But we have integrated NOH CO2 samples being collected since 2019 at the top level of the tall tower, as you can see here. So we have 14C since 2019, and since September 2021, we have also flasks being collected every week at the top level. And by the way, first year of data is publicly available in the ATO data portal. If you try to connect right now, you're going to be a bit unlucky because the server is being updated, but after October 1st, it's going to be accessible again. And just type it on Google and you can see this data. And the collection used the ICOS standard flasks, 3-liter flasks for 14C measurements, and a portable sampler, in this case, to collect from these different heights at a general cycle. So here, the results. Here I'm showing directly the millitons, for the millitons approach, but actually for the killing plot, you're going to see in the table later that it's very similar estimate. But as you can see, the isotopic signature of the ecosystem respiration in October 2019 was around 32 per mil with a standard TRO of 8 per mil. And in December 2021, actually it increased surprisingly, and it increased to 78, but with a slightly, a bit larger error of 24 per mil. Still, they are statistically different, even though the error is larger in the second one. So yeah, that actually was a bit surprising, but we want actually to first convert it into the mean transit time. To do that, we used the Corso data for some South Hemisphere stations that would fit with also the data that we have at ATO. And just smooth the data, do some fitting, and that's what we're going to use as our reference for the radiocarbon signature entering the ecosystem, and we compare that with what we measured at the ecosystem respiration. So basically, by just subtracting the year of collection from the supposed year where this radiocarbon entered the ecosystem, we get an estimate for the mean transit time of carbon. And for October 2019, it spans from 4 to 8 years, while in December 2021, it spans from 14 to 23 years. So yeah, that difference in the radiocarbon signature also means a large difference in the transit time. So let's see how it looks like compared to the other approach that I showed in the beginning. So as you can see, in terms of method, like an approach for the regressions, it doesn't have a huge difference using killing plots or millitons plots. But the main difference is between the years and slightly the months. The interesting thing here is that if we compare, if you take the turnover time in the global sense, the first approach that I showed you, actually it looks more like the results we get for December 2021. When we compare to the 7-pool model in Colombia, it's kind of in between our first and second campaign. And if we compare with the data synthesis for the Central Amazon, actually their results look more like our first campaign in October 2019. So as conclusions, in any case, we estimate that our mean transit time for this plateau area in a near pristine Central Amazon forest is between 1 and 3 decades. But the thing is that the age of the respired carbons apparently can be very dynamic, and it can change in principle between seasons or years. That makes sense, but it was not actually showed before. And that's a big contrast with actually the model approach, because the assumption is you need to assume some equilibrium, so at set state conditions, they are not predicted to change the mean transit time over time, and actually maybe this mean transit time could change over time, and that should be taken into account also in models. Yeah, we highlight some potential reasons for this variability that we observed, this difference that we observed between these 2 years. That could be simply some natural variation of the ecosystem processes due to seasonality, or maybe some specific environmental factors like precipitation. Of course, is always a very important thing when speaking of Amazon. Human activities also, such as fire and so on, and a natural spatial and temporal heterogeneity of the sources in this respired carbon. Nevertheless, these methods seem very useful in tropical forests because we have these large gradients and diurnal variation of the CO2 concentration and 14C, which brings us to very nice regressions when using end-member isotope mixing models. And as a bonus here, we did a very similar thing, a similar method for a boreal forest in Sweden, using one of the ICO's ecosystem stations, and that it was published very recently, maybe 2 weeks ago, it was public. And yeah, you can also access this through the DOI or the QR code. So almost done. So here I just want to show you one of the most beautiful things. I think it's like seeing from the top level of the tall tower, we can see these flying rivers over this carpet of forest. So these are actually clouds just flying over the Amazon. And but it's a bit sad to see that also lately probably you've been seeing the news how the forest is burning, and it's a big threat for the whole environment. And as you can see, tropical environments are really important for the global climate. So we cannot forget that, I mean, it's important to have the physical basis to understand and deal with the climate change, but it's not enough. Finally, I want to acknowledge all the people that work with me in this amazing project. So many people. Axel, thank you so much for measuring so many samples and answering all my questions. And yeah, some references, and I think that's all. Thank you very much for your attention.
Thank you, Ingrid. We have time for questions. Walter.
What is the altitude of this tower? I mean, I assume there is also an altitude dependence on this whole exchange processes.
Yeah.
And the Amazon forest stretches all the way up to Peru and into the mountains.
Mm-hmm.
Okay.
Yeah. So actually the altitude at the Ato site is not so high. It's about 130 meters above sea level. But actually indeed there is a topography difference. So we have the river and we have a slope, and above the slope we have more 100 meters until we achieve this plateau where the tower is. So in total it's about 200 meters above.
Any other questions? I have a question.
Yeah.
In your conclusions, you were taking consideration fires, which is during the dry season. But did you consider the plumes of fossil fuel emissions coming from the Amazon River? Because the Amazon River now is a freeway of shipment. And you have basically right now, anybody can Google and you will find 3 cargo ships going up and down the Amazon River. And they are burning diesel and crude oil. And the information that we get from the literature regarding the atoll on CO2 emissions, NOx, SO2, that they are capture fossil CO2 in the atoll. Have you considered that in your calculations?
No, I didn't do any correction for potential fossil fuel sources. Actually, if there is any fossil fuel, it shouldn't be so large for this kind of approach because the ecosystem would overcome, especially at the level that we are studying. But of course, it's important to take this into mind. For example, with these more frequent fires that we are having, especially in these El Nino events that are becoming more strong. We see peaks appearing in the atoll, not only from the fossil fuel, but also from the biomass burning, and especially from East Amazon, that's more degraded place. For the next, we want to keep doing these campaigns in a continuous base, so then we can actually have a real view of the variability of the years, and definitely we can try to consider more explicitly these effects. That's a good point.
All right, thank you.
Thank you.
Any other questions? Okay, let's thank you, Ingrid, again.
Thank you.
So now I am moving to me as a chair, because I'm going to present Guaciara's work. So she'll be talking about lower latitude atmospheric post-1950, high-precision tree-ring, forging sea records, challenges, and surprises.
All right. Thank you, everybody. Thank you for the organizers for giving me the opportunity to give this talk. Today I'm going to show you a little bit of my work that I have been doing for the last 5 years or so. It's a bit of the overview, so let's do it. So you're familiar with this map. It's the global map, the most recent one from Quang 2022 for the post-bomb 1950. And I had the opportunity to provide some information for the records in South America, which we have Altiplano and Camanducaia. But what we are still missing is at the tropical low pressure belt, which is related to the mean summer and winter positions. These data sets are still very little, very short. We have just one data set in the global, of course, in the global map, one data set in South Hemisphere Zone 3, and just 3 data sets in the North Hemisphere Zone 3. So my project is to build the lower latitude Cav 14 data sets. So in this project, I have chosen sites across the Amazon basin and also across the Atlantic Ocean, which show in the globe that you're seeing on your left. And I try very hard to choose 3 species coming from master chronologies which the dendrochronologists I work directly with them provided these amazing dendrochronologies. But I evaluate the calendar dates by screening them through the annual formation by carbon-14. Because in many cases, the annual formation is not quite right. So as you can see in the bottle right here. So this takes a lot of time, and this is the first stage of the project. So after the selection, I also have some other requirements. The data that I produce is evaluated with a lot of replication, which come from whole rings. And when I say whole rings, it's early wood to late wood. And when it's possible, I also have slices of the years 1962, 63, and 64, because it allowed me to anchor the records for the calendar years, for the monthly adjustment. This is associated with the rainfall regime for each given site. So I also have, for most of the time, only one single chemical extraction procedure. I'm not going to torture you about this chemical procedure. My student is going to give a little overview on that, and this is the next talk. And we also do data variance evaluation, uses several trees per site and mix 3 species per site to evaluate what's the dominant parameters. And that was shown yesterday in a poster by another student, Lucas, that is here as well. All right. So then finally, I look in the growth of my trees, taking account the precipitation, of course, because the trees is growing when it's raining during the wet season. But I also take into account the wind pattern, because that is what I take the carbon-14 concentrations to site, all right? Okay. Now with all that, I evaluate my results, and I'm going to show to you today is 3 complete atmospheric carbon-14 results. It's coming from Central and Eastern Amazon Basin, and another one from Cameroon, Africa. I had to complete another 2 from Southwestern Brazilian Amazon and the Southeast Brazil region, but I will not have the time to show to you today. So then I make comparisons with the compilations of Juan et al., and when it's possible, also with standalone records. Then I make my conclusions and comments using the tree growth and air masses, as I mentioned before. All right. So first, the Central Amazon record. This one was complete with running from 1938 to 2007. And it's coming from Porto Trombetas, which has the latitudes very close with Muna Island, Indonesia, which is the backbone of the South Hemisphere Zone 3, as I mentioned before. So I also put in a map at your left, the records of Altiplano and Camanducaia. So this record, which is showing below and in the panels, is in red, and it matched quite well with Indonesia for the rising of the Bomb Peak, as well as Altiplano and Camanducaia due to the position of the latitudes. So as the Bomb Peak progresses, the red curve starts to deviate from all others. That is related to the presence of mining at the Porto Trombetas. Porto Trombetas has a huge mine operation there, actually the entire state of Pará, which extracts bauxite from the floor. So bauxite is the main product of aluminum. And these cargo ships is what take the bauxite out of Porto Trombetas all the way to the Amazon River to the Atlantic Ocean. So consequently, this record is only good between 1938 to 1970, and from that point forward, we estimate what's the contributions in terms of fossil fuel emissions. This is only a rough estimate, of course, based on compilation and in the standalone records that we have. as 3.7 ppm, even though that is not compared with urban emissions, but it's constant because it happened during the wet season, during the dry season, during the day, during the night, because the ship, when it's in a port, is burning all the time. This was published in Environmental Research in 2022. So the second record, also coming from the state of Pará, is the Eastern Amazon base. That one is running from 1940 to 2016, but this is slightly higher than the Central Amazon that I showed to you before. So it emits the fossil fuel emissions that I had just showed. This one was complete with 773 rings and 175 analysis, lots of replicates, interlaboratorial data, which was provided by Quan, which showed to you in the right, and also slices, which allowed me to confirm the growth of the tree. So in this record, when I did train the data, I can infer longitudinal asymmetry. So we knew already that we had latitudinal asymmetries, but now we have longitudinal asymmetries as well. And the terrestrial biosphere enrichment that, thanks to Ingrid, I don't have to explain to you what it is. So from 2000 onward, we have a fossil fuel intrusion coming mostly from North Hemisphere, and it has persisted since. This record also has been published in Stotten. So now I will use the rest of my time to talk a little bit about Cameroon, Central Africa, across the Atlantic Ocean. So this one is in the North Hemisphere. So it has 73 tree rings and 111 analyses, lots of duplicates, and I had the opportunity to do the slices of 62, 63, and 64. So this tree, it grows quite slowly, and thanks to that, we can see in pink the profile of photosynthetic and respiration that's typical from the North Hemisphere trees. So it's a very interesting tree and very interesting record in one sense, because even though show that and is growing during the boreal summer, it also has a low bomb peak maximum compared to the North Hemisphere Zone 3. Why is that? All right. I compared then Cameroon with the only 3 records that I had showed to you before in that global map. So we have in the global map Ethiopia, Thailand, and India. And I put in Cameroon as the reference line in dash. So it's clearly that Cameroon is lower than all these other records. The reason for that, we have Cameroon there represented by this light profile. During the growth of the tree, we have the TLPB, the tropical low pressure belt, moving up, and consequently when the tree is growing, the air masses is coming from the South Hemisphere. So if that's the case, now we also have to wonder the contributions for the dry season fires from the Central Africa. So we went to the literature, and these plumes of Central Africa, they can be seen all the way to the Amazon base. That's a well-known phenomenon. So how much of that is going to Cameroon? So according to the literature, it's quite small. The amount will be just about 8%. So we run a model, and looking into the backward trajectories, it seems that's quite true. 5 days backwards, we have contributions come from the coast of Guinea, the Gulf of Guinea, sorry, and it will be what you see in red in the left. But 25 days backwards, we have contributions from the middle latitudes and all higher latitudes. So indeed, we have South Hemisphere's contributions all the way to Cameroon. When we compare Cameroon with Eastern Amazon basin, we see similar features, which I had mentioned before. Longitudinal asymmetries, terrestrial biosphere enrichment, and this is the first ever high-precision winter comforting record at the lower latitudes, because the tree is growing in the boreal summer, but it has comforting signatures coming from the South Hemisphere. So, some take-home message. We are able to produce high-precision records in the tropics. We had produced so far several records. We have about 600 in total measurements. 2 records from Amazon basin has been already published, and these newly developed 3-ring records at the lower latitudes allowed us to infer asymmetry pathways of interhemispheric air masses, biosphere-atmospheric interactions, and fossil CO2 contributions, locally from mining or non-point sources. Based on the Cameroon CAR-14 record, South Hemisphere winter CAR-14 levels near the equator have been detected for the first time. So I leave you with some words from late Ingeborg Levine. It's true, she agreed that modeling was great, but they cannot replace observational data. And that's what we are trying to produce. Finally, ta-da! I can't compete with raw talent, sorry. A special thanks to my students, they are here, June and Lucas, and all of you. And I'm open to questions. Thank you.
Thank you, Guaciara. So we have time for questions. Ingrid?
Hi. Thank you very much. It's a very nice talk. And I think, I don't know if it was clear for me for the slices of 3 rings, how exactly you slice them? Is just by the patterns, or you also performed like 18-0 measurements to see this interannual variability related to precipitation?
Oh, that's an excellent question. It's not possible to see any pattern like if you're asking like to see early wood, late wood, that's not possible. I barely can see already the boundaries of separating the 3 rings. June will show you some of the, at least small images how some of these 3 rings look like. They have lots of parenchyma, so it's very hard to separate them. So we use the technique that was already shared with us from Peter Grutz, that used just a division of the 3 rings. So we choose by mass, yeah. Okay. But it's very, like, try to be very precise. And we try to not lose any material. So the handling in the lab is very critical. We have basically 97 to 100% recovery yield in the cutting. So we are quite critical about that. If I had the chance to maybe show the, to avoid what I call the yo-yo effect, which make results like in a rising to change, you know. So one thing is, if you want to just confirm if, for example, a tree species is annual or not annual, that phenomenon from 1963 nobody cares if it's going up and down, but the truth is the tree cannot be growing in 2 different growing seasons, right? Because that is what's showing right there. You know, all the trees grow in a wet season, or they grow in a dry season. Cannot be, right? But that's not the fault of the tree. It's because who was cutting the wood lost material. So to avoid all that, we are very careful when we are sampling the wood and when we are cutting the wood in the lab and then later chipping the wood and so on. We have all these sorts of strategies, but anyway, this is all already published, you know. But I call that yo-yo effect. You make the thing go up and down in a 63, 64, they go all over the place. Yeah.
Thank you.
Another question? Walter?
Thank you very much for this interesting report on Cameroon and Africa. I'm just wondering whether you plan to extend your measurements also into the non-tropical forest areas and in areas where the tree growing is much more sensitive to climate. As far as I remember, there was recently a very surprising collapse of the boab tree in several places in Africa, and one amendment doesn't really know whether it's related to a climate change, but does the tropical forest tell you much about climate change or not? I don't get this message. Is it rather, I could imagine that the drier areas are more sensitive. I mean, measuring trees in dry areas, yeah, okay.
To be honest, I'm very open to collaborate with other species or if somebody here has the material, but I have been talking to Lucas about that. Yes, the answer is yes, I would like to. I have pretty much exhausted all the sites I want to work in the Amazon at this point, and I want to move towards Africa and Asia, and we have been talked to some folks here, but it's important to work very closely with the dendrochronologists because this is so critical, this portion here, this work is so critical. So yes.
Okay, so let's, are you singing? No singing? No singing. Okay. So thank you for a nice presentation. We are behind time, so we get the next speaker then.
Thank you.
So now we have Jay Griffin. Hello. Talking about evaluating a simplified alpha cellulose extraction procedure on pantropical woods via FTIR characterization for use in atmospheric carbon-14 reconstructions. So you have 20 minutes that we will start soon.
Hello, can you hear me okay? Good morning, everyone. My name is June Griffin, and today I will be talking about my research using FTIR characterization of pantropical woods and their alpha cellulose, as well as its implications for past atmospheric carbon-14 reconstructions. All right, so past atmospheric carbon-14 reconstructions are crucial for describing the history of the Earth's climate. Carbon-14 datasets serve as a baseline for a variety of studies, including better understanding air mass mixing and dynamic climate changes, air-sea flux of carbon dioxide, soil carbon stocks and turnover times, and ground-based fossil fuel carbon dioxide emissions, as well as sources of carbonaceous aerosols, many of which we have had the pleasure of hearing about during this conference. So this study is part of a larger project to bolster the carbon-14 records of pantropical regions by verifying that tropical trees can be efficiently processed and dated. So we all know that tree rings are important for radiocarbon calibration, despite the majority of trees being located in the tropics, as you can see in the dark green color. Tropical tree ring data is particularly sparse. This is a map of tree ring data documented by dendrochronologists, and this is also the same spread of data from which data is taken and used to create the calibration curve. This data gap can be caused by a variety of factors, including the expenses and difficulties of collecting samples from dense forests, the high decomposition rate due to the humidity, and the high diversity of ring boundary anatomies, as well as the misconception that tropical trees do not produce annually resolved rings. So we know that wood is important for radiocarbon dating and that the data we have is currently uneven. However, there is another large issue when working with wood. Researchers have found that wood contains both desirable carbon, which we will be referring to as alpha cellulose, and undesirable carbon, which includes extractives like fats, resins, and waxes, as well as hemicelluloses and lignin. Desirable carbon is great because it gives us accurate carbon-14 readings. Alpha cellulose is a chemically derived and reduced form of cellulose, in our case taken from tree rings, which will provide the most accurate carbon-14 results for atmospheric reconstruction, which requires high precision. And it looks like this fluffy, white, fibrous material. However, mobile or undesirable carbon can interfere with carbon-14 dating. So, trees are very clever and they can store excess carbon for a rainy day by reserving it further into their body. And when a tough year rolls around, they can pull that carbon back out towards their developing rings. However, this means that carbon from different years can travel across tree rings. And this can lead to incorrect readings, such as this false bump right before the bomb peak that should not exist and can vary in age range. This is why it's so important to chemically reduce our wood samples to alpha cellulose. Our chemical treatment of choice is Santos et al. 2023's treatment, and we will make a note that it does not use any organic solvents. Now, the organic solvent step traditionally is used before the acid-base acid treatment, and it is meant to dissolve extractives. Because pantropical trees are known to have a higher extractive content than its extratropical counterparts, the success of this simplified treatment would bolster the idea and claim that tropical trees are not particularly intimidating to chemically treat. Now, this diagram to your left is a brief overview of this chemical treatment, and the light green boxes represent where we took subsamples after each step of the chemical treatment. These subsamples were dried and rinsed, and then treated with Fourier transform infrared spectroscopy, or FTIR, to draw conclusions. And you can find more detail about this chemical treatment in our 2024 publication in Stoughton. Right, so for our research questions, we were wondering if 8 tropical tree species sampled from South America and Africa could be reduced to pure alpha cellulose using our simplified chemical treatment. And if tropical trees are chemically very different from each other as suggested by their varying appearances, colors, higher extractive contents, and variations in their tree ring boundary anatomies. And what I mean by this is you can see P. paniculata, which is this vibrant purple colored wood, and just above it is Cedrela facilis, which is much more muted and light brown. So these drastic differences in color indicate that there could be something chemically different between the tree species. And both of these questions were answered using FTIR analysis, which is a qualitative method of determining the presence or absence of compounds inside of a sample. So this is the raw FTIR spectra. This is of untreated wood. Now, we took a total of 56 FTIR spectra for this project, but we will be showing you a representative 3 for clarity. To orient you to this graph, each squiggly line represents a different tree species, and each peak corresponding with a wavelength represents the presence of a certain compound. So as can be best seen here and here, despite these woods looking very different, they actually have very similar spectra indicating similar compound composition. And then we have alpha cellulose. So this is the spectra of the same tree species. And as can be seen here and here, they are also adequately similar to each other and, more importantly, match the established spectra for alpha cellulose in literature. So we found that despite the woods looking very different and having higher extractive content, they were not very different from each other. There were mild variations in compound composition, but overall they were quite similar. And this is great news because it means that tropical trees should not be particularly intimidating to chemically treat. And our results confirm that our treatment produces pure alpha cellulose extracts, indicating that this can be used safely for carbon-14 atmospheric reconstruction in pantropical areas. Now, to further verify this claim, we compared our data to the spectra of C3, which is an internationally recognized standard. That's going to be the dotted line at the top of this graph. Under that, we have EU in yellow, which is representative of our tropical samples, then Fury F and Fury H, which are both going to be reference materials and are extratropical. EU, Fury F, and H were all reduced using our chemical treatment to alpha cellulose, and you can see that they are adequately similar to the spectra of C3, with the exception of them missing the 6th and 18th peak. These peaks correspond to the presence of lignin and mannan, which are undesirable carbon, and so we can conclude that our chemical treatment can reduce samples to the cleanliness of C3 and possibly even more. And to confirm this again through radiocarbon readings, we took the carbon-14 readings of several of the tropical species here, and we also use the exact same chemical treatment method. And as you can see in this region, there is no false bump before the bomb peak, indicating that there are no remaining mobile carbon left. And this dashed red line is just an approximation of where you could see that line. This data was presented yesterday by Lucas in his poster and is also in our recent publication in Radiocarbon, which just got accepted. All right, so I also took the liberty of pursuing some side quests during this research project, including finding the purpose of each step of the chemical treatment, looking at the same species in different locations to see if environmental influences could change the compound composition of a tree, and looking at the same species in the same location to determine if there were individual differences in compound composition. So this is a very brief and oversimplified overview of which wavelengths correspond to what compounds. All the way to your left in yellow, we have resins, followed in red by aromatics, lignin, and hemicellulose, then cellulose, and finally mannan. We will be using this scale to look at the FTIR spectra before and after each chemical treatment. So here's that raw spectra again, and the acid-base acid treated wood. If you look over here, you can see that a peak goes missing. The wavelengths of this peak define it as resin. And the same thing happens here, and this peak is defined as mannan. So we can conclude that the acid-base acid step removes both resin and mannan. Then we have the acid-base acid spectra again compared to acidified bleach to achieve hollow cellulose. You see in the central region that several peaks go missing, which are defined as aromatics, lignin, and hemicellulose. This means that the acidified bleach step to achieve hollow cellulose removes lignin, aromatics, and hemicellulose. And finally, that hollow cellulose graph again and the alpha cellulose spectra. Along this slope, you can see that one peak goes missing, and that is actually representative of lignin specifically. So the delignification step to achieve alpha cellulose removes lignin. We took 2 samples of Cedrela facilis, approximately 2,000 kilometers apart, one in a wet forest and one in a dry forest. And our reasoning was that looking at samples that are very, very far apart could mean that they grew up in different soil types and under different precipitation patterns, and we were wondering if those environmental conditions could impact the chemical composition of the trees. So in that red box, you can see that there is a mild variation, but this is too little data to draw any conclusions. We did the exact same thing with 2 species of Cedrela odorata, and this time it was 1,500 kilometers apart. Again, you can see some mild variations in the spectra, but nothing enough to draw conclusions. The important point here is that despite these trees growing up in potentially different environmental conditions, both sets were reduced to pure alpha cellulose using our chemical treatment. And then we did two samples of the same species, Cedrella facilis, in the same location, and we found no discernible differences in their spectra. Again, this is too small of a sample size to draw conclusions. So now that we know that a simplified chemical treatment that does not use organic solvents is effective on tropical trees, researchers can save money on buying equipment and chemicals, time, which makes the radiocarbon calibration process more accessible and efficient, and we hope this will encourage researchers to start filling in the data gap in the tropics. To sum it all up, the Santos et al. 2023 Simplified Chemical Protocol was proven effective on 8 pantropical tree species via FTIR. I would like to give a special thank you to Gua, Lucas, the NSF grant for funding, and to the conference organizers, as well as all of you for this wonderful opportunity to learn. Thank you all for your attention. Any questions?
Thank you, June, for such a nice presentation. So we have time for questions. Ingrid?
Sorry, I think it's because I'm in front. I want to ask questions. Thank you very much for this super nice presentation. So I was wondering about I don't know if you already have some radiocarbon results, but how would be the radiocarbon results for the trees that are from the same species but different locations? And I ask that because there is a very recent work. It was just only in the Amazon basin, but the same tree species was analyzing different environments, and there was some kind of evidence that also not only environmental factors could influence the results, but also the management of the forest by native populations, for example. And I wonder if something similar could be included or discussed in your work. Thank you.
That's very interesting. If you have that paper, then I would love it if you shared that with me afterwards. We did not take the carbon-14 results of the specific samples we took the FTIR spectra of, and this is because to run FTIR spectra, you need quite a large quantity of wood, and to do multiple runs of it, in case there were any human errors, we wanted to collect a large amount, and this means that just one tree ring wasn't enough. So we merged multiple tree ring samples after grinding them down to a powder, which means that we could not carbon date them. However, we do have the carbon-14 results in that radiocarbon publication that's upcoming, and Lucas also presented on it yesterday. So that is the same species with the same method, but that wasn't in the scope of this study. Thank you for the question.
Thank you.
Guasiara, you want to?
It's just a comment. I'm guessing that you're talking about the Bertiolia Excelsior. I had it tested that the 3 species before, and it fails every single time. It's not really annual across Amazon basis, so I'm not surprised that actually was, it could not be used for reconstruction. So I dropped it a long time ago, many years ago, and I present that in AGU. So I know the papers say that it's related to some soil differences, et cetera, but I don't know that, but it's not going to be useful for reconstructions, so.
Okay, so one just quick question, because we are late for coffee break, which is very important. One there, please.
Thank you. Very good presentation and very good result. One thing I may suggest, maybe you go a little bit further, and that's you show all the FTIR is, excellent. But if you can do the measurement of carbon-14 or the different like ABA, BABAP, or, Holocenslo, and the alpha cellulose for a series of samples, and then I think it's maybe more convincing for more people, right?
I agree, thank you. If I can ever get my hands on larger tree rings that would provide enough material to do that, then I'm sure that that would make a very convincing argument. So thank you.
Okay, thank you very much. So let's thank her again and thank all the speakers of the session. And now we have coffee break, right? Okay.
start presentation.
Where? Just
I
with-
start the countdown.
You start. Okay.
Okay.
No, okay, great. Thank you very much. So we miss one presenter. I'm just gonna ask you. I don't think it's 7 minutes. Thank you.
draufstelle?
Das ist gut. Da kannst du mit deinem Bauchnabel spielen. Also ich bin nicht so, das ist gestern nicht so gut verstanden. Yeah, Ronnie was saying I could speak with my belly button. Ist nicht gut heute. Lange? Ja. Für mich ist eine Stunde. Also ich habe eh nicht getrunken. Das ist schon mal easy, aber es ist mir nicht so gut. You need it for your introduction?
No. We don't have this person. It's named one.
So we have time?
Yeah, I think so. I think. I'm not sure. No, it's okay.
Jetzt halt nicht so gut.
Vielleicht geht's in einer halben Stunde besser.
Vielleicht geht es mir eine halbe Stunde besser, aber normalerweise bin ich ja nicht so der nervöse Typ, der die ganze Zeit auf dem Klo sitzt. Mal gucken.
That would be nice.
Sta in arrivo. Ciao Resp.
almost completely.
Maya said she has to be, she's in hurry actually, so we have to start.
She said that she needs to get the trainer.
We have one, so we have to be very fast. I don't know. I asked somebody to let her
Pardon? I asked her, she said it's fine. I asked her if she wants to go first.
Okay, hello everybody. Hello everybody and welcome to the last session of this conference, and this will be the marine environment section part 2. So the first presenter is Manuel Ruben with his work The Age Predicament in Microbial Organic Matter Degradation. Please, Manuel.
Hello. Yeah. So generally I start my presentation with asking people in the crowd if they think that age is important, but obviously I think you all do. But I would like to ask you to think about something different if you think that the age of organic matter is important in the context of its bioavailability. And today I will present to you basically the summary of some of my PhD work and one big question that came out of it for me in the end. So there's also something different about my talk today. Due to the setup of the room, I decided that I will have subtitles, not headlines, so hopefully even in the back you can see my figures better. So when we talk about ancient organic carbon, it's important to understand that we as humans put a lot of it into the environment. Due to our actions, for example, due to the way we perform agriculture, due to industry products, the entire petroleum industry, but also passively by raising the global temperature, and that's particularly true for the high latitudes, what we will have a look at today for the permafrost environment and also the glacial environment. And what I asked you earlier already is many of you may know that the organic matter in permafrost deposits is considered to be highly bioavailable. Whereas the organic carbon in the bedrock generally is seen not to be bioavailable because it was degraded already a long time ago and for many, many millennia. And well, that's what I looked at because we have increased rates of the input of these organic carbon substrates to the environment. And well, I think I have to step aside. The question is, if they get degraded, do we get additional carbon emissions from those substrates, which would in turn then increase global temperatures again and cause a positive feedback loop. So how can we figure out if microbial organisms, for example in sediments, I think I will step over here, can utilize organic carbon from different pools? Well, for example, if we take a sediment and there are two different sources for substrate, for example, phytoplankton and coal, they both have an isotopic signature. If there is a microbe that comes along and it feeds on the organics in the sediments, it produces metabolic products like the greenhouse gases CO2 or methane, depending on the setting, but also, for example, dissolved organic carbon. And so this is basically the catabolysis. For the anabolysis, it's the same, so they build up tissue. So when they feed on some type of organic carbon, they implement the same isotopic signature in, for example, the cell membrane. And when we use chemical purification methods, then we can obtain in the end the isotopic signature of both the product, the metabolic product, as well as the tissue. And today we will focus on the lower one. Basically in this example, we have 50-50 from both sources. Well, today we focus mainly on the membrane lipids, especially phospholipids, as they're an indicator for living microbiota, because the head group, which you can see here, when it bears phosphate, it's very prone to degradation and can get lost within days to weeks after cell lysis. So let's have first into the permafrost environment. So there have been many studies concerning permafrost organic carbon lability in the terrestrial realm, but not as much in the marine realm. But the Arctic coastline alone stretches around 1/3 of the global coastline. So there's a lot of exposure to rising sea levels and rising sea surface temperatures. So there's a lot of erosion happening. And to my knowledge, there are 2, maybe 3, depends how you count this one study, studies that look into the remerization of the organic matter directly in the water column. And but there is no direct work that quantifies how much of the organic carbon gets actually remineralized once it settles on the ocean floor, which is, you see here. And what we know, or what the numbers that we take, are generally derived from proxy work. And that's what we had a look at working on sediment cores. We used for that sediment cores close to Herschel Island, which you can see here on the bottom left. It's west of the Mackenzie River Delta in Yukon, Canada, at the Beaufort Sea coast. And you can see also indicated in the colors here that there is quite a bit of coastal erosion happening in this area. Additionally, in yellow, you see retrogressive salt slumps. We saw them already day before yesterday, and I think day before yesterday. So there's a lot of material get put into these sediments locally, and we will focus on the sediment core, the magenta colored one for today. So this looks a bit busy, but basically what we did is we took both the 13C and the 14C isotope to figure out where, first of all, comes the organic matter from in the sediments. And we have 3 end members. So one is the permafrost that has been frozen for thousands of years. Secondly, the active layer here with AL. So basically the zone where you have annual thawing and freezing cycles, and also where there's still fresh vegetation is growing. And then we have marine primary production on the top right, so basically phytoplankton that grows in the sea. And first of all, if we plot the content of the isotopic signature of the TUC in this diagram, then we see it falls basically in between the permafrost organic carbon and the active layer. So, like I already said, there's a lot of terrestrial material being deposited there. So, let's see here, it's about half active layer and permafrost contribution, and only about 4% come from marine primary production. However, if we look at the isotopic signature of these membrane lipids, so basically what the microbes feed on, we see a very different picture. So it moves very far to the right, so much younger material they actually feed on, and it's also isotopically heavier. And in terms of numbers, when we basically calculate how much they feed on, is that they nearly use 80% of their carbon for their biosynthesis from the phytoplankton that gets deposited in these sediments. The same is true if we look at the branched 15O fatty acids. then it basically shows the same picture. And then you say like, oh, maybe they feed on it, but it might be not the same that actually comes out of the sediments. But we also did the same on DAC, so dissolved inorganic carbon. And we heard earlier today what a Miller-Trans plot is. Basically, we did the same to figure out the source of the DIC, so what gets produced in the sediments. And here we see a bit of a shift to more the permafrost and the active layer contributing to the DIC that gets released, but it's still a lot of the marine primary production, and a lot of the DIC is bubbling out from the deeper layers, so maybe there is just a shift due to time. Anyway, this shows a very different picture than what you would expect when you read about permafrost in the literature. So you think like all this stuff should be readily available and be degraded when it sits there. But yeah, it's not so biolabile as one would think. So that was very surprising for once. The second project, I'll have to look at the time, it was in the high Arctic, also in Svalbard, on Hornsund Fjord. This is not Hornsund Fjord, but the picture really nicely illustrates what I want to show you. So in brown, you see ancient organic carbon that is transported through the glacial runoff into these fjord systems. So this is one of our end members. And the second organic matter source is modern organic carbon, as you can see nicely here in green, produced again in the watercolor. We did that on 3 cores in the fjord, and please remember the colors. In blue we have the core that is most marine influenced in the main basin, and then you see in magenta and green 2 cores that are located in the meltwater glacier dominated Brepollen basin. So you have a big sill around here, and this is all dominated by the glacial runoff. So basically same scenario as before, however just for 2 isotopes, so for 2 end members. So at the left side you see always the depth of the core, and on the left side, so at 0, you would have the isotopic end member for the ancient carbon, so for the petrogenic carbon, which is obviously 0 because it's there since millennia. And then in yellow, what the phytoplankton would have, judging by the DIC, by the local DIC in the seawater. And in black, you see the total organic carbon content of the sediments. And as we have basically an end member of 0 and 1, we can read it also as percentage. So here we see that in the outer fjord, about 45% at the top, down to about 25% at the bottom comes from primary marine production. And the rest is from the petrogenic organic carbon. So this is how the outer fjord looks like. If you look then onto the core closest to the glacier, then we actually see a very different picture, is that we have much, much less organic carbon derived from marine primary production, so only around 5%. And the same we see if we look deeper in the sediments with the long core. There we start around 10% because it's a bit further away from the glacial termini, and it goes also down to about 5%. So yet again, when we plot into that the isotopic signature of the bacterial membrane lipids for the outer fjord, we would see what we would expect, that the microbes primarily feed on the fresh material. However, we have a bit of a divergence in the lower parts of the core. If we then, though, look at it at the glacial termini, or terminus actually in this case, we see quite an offset to the yellow line. So at least 2 parts, the microbes use old material for the biosynthesis. And the same is true if we look further into the deep core, and we can yet again look at that in numbers. So in the outer fjord, in the topmost, 15 to 20 centimeters, only about 10% of the carbon they use, but I think it's still quite a bit, is from the ancient carbon. Whereas if we go closer to the glacier, it increases rapidly, and we get up to 56% of ancient carbon contributing to the microbial biomass in these membrane lipids. So this is very surprising, because you would think material that was synthesized and deposited in the tertiary would be biologically inert by now, because everything is thermally or microbially utilized by now. So this brings me already to my conclusions. So number one is that petrogenic organic carbon does not necessarily have to be biologically inert, which is quite surprising. Secondly is that at least the particulate fraction of the permafrost deposits, when they settle on the ocean floor, are not as biolabile as we think, or at least the marine primary production is consumed primarily, or the organic carbon from marine primary production. And third, I think this is my key take-home message for you, is like drawing conclusions just based on the bulk or the source age in terms of the bioavailability of organic matter. It's, in my opinion, insufficient. And I think that's what my thesis showed. And the question that for me arose is, do we really understand microbial ecology that well in terms of organic matter age and what they can actually consume? Okay, thank you. So I would like you to help me with that. So, volare, oh, cantare, oh. Thank you.
Okay, thank you, Manuel, for your presentation. It was very nice, and also thank you for your excellent time. And so we have time for questions, if there are some questions. Do we have questions? Oh, it seems there are no questions. So, okay, we can start earlier with the second presentation. Thank you once again. Okay, so we move on to the next presentation. The presenter will be Bente Philipsen with the lecture, Human Environmental Interaction in a Prehistoric Fjord. So, Bente, please.
Thank you very much. So let's see. There we are. Yes, I'll talk about how humans and the environment interacted with each other during the Stone Age, and especially how radiocarbon dating can help us to find that out. It's a part of a research project that is ongoing, and I don't have many results, but this research project is based on a huge excavation. And we move many hundreds of kilometers north from here. So we are on the south coast of a Danish island, where due to development works, 300 hectares of rescue excavation needed to be carried out. And fortunately, almost 190 hectares of that were wetland with excellent preservation conditions. So even though we were only able to do the rescue excavation and collect material and a bit of data, we have this wealth of material available for us now. And that's where the first research project kicks in, Sylfish, because the fjord was called the Syltom Fjord, and it's about fish. And the environment in this fjord was changing throughout the human habitation in the area. And based on the data from the excavation we have so far, we reconstructed a sea level curve, where we did a very simple model. We assumed that a fireplace or a living tree were above sea level, and things that had been deposited in the water, like fish traps, they were below sea level. And we just drew that onto a graph like this, and then just by hand drew a line through it, and that's the sea level curve for that region. And in contrast to other areas in Denmark, there was no fluctuation, so not going up and down, but a steady increase in sea level until today. But something else happened, and quite dramatically. Around 4000 BC, agriculture finally reached the region, and we have, in the traditional view, there's a whole package of things arriving. We have the domesticated animals and plants, and we have a cultural change. I won't go into discussing the term of culture, but we can see that the pottery shapes change a bit. And there was a mouse pointer before, but you can see it on the screen. So we have one type of pottery before 4000 BC and then another type after. And now I want to look into the details. How did this transition happen? Because we have this wealth of material on the site. And the first thing we did is we radiocarbon dated all the stationary fishing structures. People had built fences into the fjord to guide fish into traps. I mean, it's much more convenient than standing there and trying to catch them actively. And that had been going on for thousands of years, and also at the same locations. So even though the sea level was rising, the fishing didn't really change much in terms of, for example, the location. And if we, for each excavation trench, sum the radiocarbon dates of those fish weirs, we see that there's a long tradition, although we have some phases with where they were more actively building those structures. And when we compare that to the rest of Denmark, you have in black the sum of the sums, which I just showed, and in gray you have the Danish data for comparable structures, like fish traps. And in blue we marked where we have fewer fishing structures in our site than in the rest of the country, and in red where we have more. But what is probably most interesting here is that we have one peak around 4000 BC and then one peak later, after the introduction of agriculture. And another traditional view of the transition from the Mesolithic, where people were hunting and gathering, to the Neolithic with the agriculture, that is that the diet changed totally. So this is from the first publication on the subject in 1981. We have the radiocarbon dates calibrated on the x-axis, and on the y-axis you have delta C13 values, which show that until 4000 BC people had a completely marine diet, and then on the day, 4000 BC, they shifted to in the Neolithic to a terrestrial diet. And this is an ancient publication, but the general picture is still true. So we have, when we go back, intensified fishing, and at the same time a total shift in diet away from marine resources, which is apparently a contradiction, but I will show later that it isn't. So what have people been doing around the transition to agriculture? Well, they were hunting wild animals. Here's a sum distribution of 74 dates of wild hunted animals from that site. And they were hunting them quite intensely. There was an intense habitation also around 4000 BC. But they never really stopped hunting animals, although they were domesticates. And I also did a correction for the reservoir effect, which is not totally easy because we have the Baltic, which is not the global mean ocean. It's more like a freshwater system because there's so much freshwater runoff, but you need a reservoir effect. So I did a very simple assumption based on some marine and terrestrial paired samples that we just correct with 250 years. Doesn't change the general pictures. I mean, some wild marine animals were moved a bit, but the general picture is still true. Now, when we take a look at the domesticated animals, put them at the top, they appear, well, around that traditional transition at 4000 BC. And you can also see that there's a decline in hunted animals before the peak of domesticated animals starts. And when we just take a bit closer look at less chaotic look at those dates, so I used the KDE model just to get rid of the wiggles from the calibration curve. You see that there's first a peak in wild animals and then in domesticates, but people never really stopped hunting, and there's even a small peak afterwards. But you have to be a bit careful with those domesticated animals. They were all identified by osteologists. And here's, as an example, I took the sheep and or goat bones, which were radiocarbon dated, and also with an overview distribution at the top. But when you then take a very close look at each of those individuals, and you could use something like Zoom, so zoology by mass spectrometry, where it's basically the protein analysis. And the proteins, well, the different amino acids in the collagen depend on the species. And when you do that, you can get some surprises, which means the first four are okay, so 1 goat and 3 sheep, but the rest were misidentified, so they're actually roe deer and even 1 red deer or elk. Which means also that the distribution shifts a bit, making the introduction of the true sheep and goats a bit later than the first one. So my previous figure with when domesticated animals were introduced, to have to be a bit cautious about that. Maybe some of the cattle or sheep were actually wild animals. But let's take a look at what the people actually did with all those resources. So here I have a graph of the C13 and N15 values of food remains, just charred food. You maybe have experienced it yourself, that when you're a bit unlucky and you don't turn down your campfire fast enough, then the food can burn and char. And you see that there's a great variety in resources, and this is only the Mesolithic, the hunter-gatherer pottery. You have both terrestrial and marine C13 values, and also the N15 values, so all the way up the food chain. And written in text is results from lipid analysis of the same pots. So these are not full quantifications, but they are biomarkers. That means that, for example, if I write marine, it means there has been some marine fat in this pot. Doesn't mean that it was like only for cooking fish, but there was something marine in it. And well, you have this variety, which makes sense for hunter-gatherers, and especially when they were fishing so much. And now the traditional view would be, with the introduction of agriculture and human bones show clearly that people were only eating terrestrial food, we should expect all the pottery from the next phase to be in the terrestrial corner. But it isn't, not at all. You have the same spread, or the same variety of different resources that were used. What is new here is that we have some clear markers for dairy products. So although people were still using like the same variety of resources from the land and from the sea, they also used dairy from the very earliest times on. And now we would have liked to show you some data on human remains, but there were no graves at that site. So the only trace of a human that we have is a piece of chewing gum and the DNA extracted from that. And of course, the DNA people can tell you a lot about the phenotype of that person, and that's how she was reconstructed. I must say that we don't know about her age, so that is the artist who thought it would be nicer with a young lady. Although there are no tooth marks in the chewing gum. You can see that it had been chewed, but no distinct tooth marks, so maybe she wasn't that young. Anyways, the radiocarbon date of this chewing gum is 3,800, maybe with a small peak at 3,900, but after the introduction of agriculture and after the immigration the possible immigration of people with a different ancestry. So this is, especially with that phenotype, still the Mesolithic, the hunter-gatherer ancestry, which persists for quite some years here. So if I do a very preliminary summary, we see that from 4000 BC we have a new material culture. And when you have a very close look at the pottery and at the flint, you see that there's both a continuation of local traditions and you have some impulses from the outside. And once the first domesticates arrive at around 4000 BC, not earlier, those were misidentified, there's also dairy already present. And people couldn't digest the lactose at that time. That's a mutation that only appears much later. So they were not only able to have cattle and use the milk, they were also able to produce something from the milk that they were able to digest. Otherwise, we wouldn't find dairy residues in the pots. So although they were still hunting and gathering and all that, and especially fishing, they were already making cheese as well. And from DNA studies, we know that there's a population turnover in Denmark at around 3800 BC, so there must also have been some kind of immigration going on. And what we find not only on our side, but throughout Denmark, is that there was intensified fishing, especially with those large structures where you can fish passively. So based on all this data and on digging in the mud at that site for years, I have some hypotheses, which may or may not be proven right after our projects here. So my hypothesis would be that the inland areas, which we didn't excavate here, were settled by incoming farming populations who brought all this technique of keeping cattle herds and using dairy and producing stuff from it. They were in the inland areas, and then on the coast, we still have the hunter-gatherer population, as you also could see by the DNA analysis. And I think that there was an exchange going on, which leads to dairy residues in the pots on our side, and which also sparked an increase in hunting and fishing activities, because when you buy the cheese, you have to pay back with something, and that could have been fish or, I don't know, what people would have liked to get in exchange for the dairy. And this exchange also led to the remains of domesticates at the coast. So maybe the local farming population just traded a few cattle and a few sheep and goat, and the hunters weren't like totally switching to becoming agriculturalists themselves. And I think that this intensified fishing is not visible in the human bones because we had the population growth. So I think that once you have the farming populations coming in and settling areas that were unsuitable for hunting, gathering, and fishing, that you have a population growth. And although there was an absolute increase in fishing, there was no relative increase because you just had more people and producing more terrestrial resources. And of course, these are just hypotheses, which make sense from the data we have now. The challenge would now be to test them. And how can you do that? I have one tiny preliminary test where I just looked at a few animals from a restricted area. Here I've split them up again. You've seen it before with the domesticated versus wild animals. And now I split them up into animals you would hunt for their meat, like roe deer and red deer, and fur animals, like wildcat and otter and those. And I also treat the marine animals separately because I think that oil from marine animals would also be good for trading. And here we see that there's a peak in fur animals exactly at the time of transition. So my hypothesis would be that people intensified those kinds of hunting activities that would be good for trading, because fur, well, you need it if you've ever been to Denmark. You need some warm clothes. So I think that the incoming farming population would have been happy to trade fur for dairy or domesticates. And I think that this is already a hint that there's some change going on in the local hunting strategies due to impulses from the outside. But we've only started with our research project, so there will be more tests of this hypothesis. And Especially we are focusing on the fish, so we will also have a look at did they target different fish species and how did the isotopes of the fish change? Did the environment change when the fishing was intensified? And so on. So maybe in 3 years' time, I'll present all the final results from that project. So I would like to thank all the colleagues who did the excavation, and of course my colleagues in the research project Silfish and at the local museum and Augustinus for making it all possible. Thank you.
Thank you very much for your nice presentation. Now we have time for 2 questions, maybe 3. Are there any questions? Okay, everybody would like to go home. Okay, thank you very much. Thank you very much once more. Okay, so we move to the next lecture, but it seems that we don't have one presenter, so we will just skip it and proceed with the lecture of Maja Alsicek. So Maja is here. Presentation, nanoplastic uptake, dynamics in muscles, unveiling first sub-PBB toxicokinetics. Maja, please.
Okay. Good morning, everyone. Thank you very much for welcoming me in the Radiocarbon community, because as you can see, I'm not someone who use AMS like you do guys for datation. So I'm associate professor at the University of Edinburgh. I started last June, so I'm in a transition. But what I'm presenting today, it's a work that I did with ETH. Because they did pioneer some really nice work on the AMS, which allowed me to use it. So I would say that I worked on plastic since more than 30 years, I would say. So when I was a little girl, every day over the summer, I was going on the beach. And I was collecting those plastic on the way because, you know, that was everywhere at the time. My mom was really mad because she really thought that I will get any disease that was, you know, on those plastic. But in my head was like, well, those plastic are there and they will never disappear. So as a little girl, I thought that the magic box was to put them in the bin, but they do not disappear still. So imagine that I fell to my purpose of cleaning the environment, because currently we have above 8 million tons of plastic discharged every year into the ocean. So this led to things like this turtle, like you can see, entangled in plastic litter. So it's quite emotional, this physical damage, and animals die because of, you know, being killed by those plastic. But 20 years ago, a marine biologist realized that actually this plastic litter was degrading and fragmentating into what he called microplastic, but you have to excuse him because, you know, it was particle below 5 millimeters and he called that microplastic. So if you want to be really accurate, no, it's not very micro. Anyway, recently we also realized that this fragmentation, because of the weathering, is not stopping, obviously, and we have those particles fragmentating further to reach nanoplastic sizes and what we characterize below 1 micrometer. So as the size decreases, you can't see them, but the property will change and their impact might change. And the question is that those plastic particles that we can't see are a problem. So the EU did a report in 2019 mentioning that potentially nanoplastic is the most hazardous form of the plastic litter, and we need to investigate what is the impact of those particles. So obviously, a lot of people started to work on that and led to some debate in our community, dividing between people saying that, well, you know, it's very impactful, so we really need to look at this problem of plastic litter. But other people were saying, well, no, it's not a such big problem and we should not be caring about it, really, and you should be giving more money for doing something else, you know, for the funding. But This debate started by the fact that those sounding the alarm were using very high concentration of plastic for performing ecotoxicological tests. Because when you perform an ecotoxicology test, you want to see a biological response. And to reach that biological response, they had to use high concentration, so above the milligram per liter level. However, recently, a team detected, I would call nanofraction, because they did melt an ice core, filter the water from this ice core, and detected below 200 nanometer some polymer. And this concentration was about 13 microgram per liter. But no, ice core is a sink environment, so you will concentrate those plastic. You can imagine that in the open ocean, the concentration will be even lower than 13 microgram per liter. So a few years ago, when I was looking for a postdoc, I applied on a postdoc with Richard Thompson, the marine biologist who realized the microplastic was produced from this marine litter. And I've been a bit bold, because I arrived saying that, well, you want to look at the uptake of those plastic particles that we can't see, and you want to see how it behaves in an organism. So the best way to go to work at environmentally realistic concentration is to use the radiocarbon, because of the abundance of the carbon. So using nuclear technique like liquid scintillation counter and autoradiography, I've been able to reach the 15 microgram per liter, which is the upper limit of this environmental concentration. So what I did in that experiment was to label polystyrene, which is the third most abundant plastic produced worldwide. And, being able to incorporate this carbon-14 into the polymer, I exposed some scallops, which are a filter feeder, meaning that we will look at the absorption and the depuration of it, and also being able to quantify for the first time in the field the residence time of those particles into a body. So I did expose for 6 hours at this 15 microgram per liter. So we saw an uptake about 30% and a depuration happening very quickly, because within 2 days, 80% of those particles was depurated. But it's a laboratory-based experiment. This is a pretty, perfect environment in the sense that I expose the animal, and after that I transfer them into a clean water. But in the environment, that's not what's happening. They are constantly, chronically exposed to those 15 microgram per liter. So using the parameter that I've been able to extract from that quantification, I've been able to model and to predict how much of those plastic particles will be accumulated over a year. And I will stress that I did quantify into the tissue. So you see on the left side, you have the bioconcentration factor model, and on the right side, I did convert into a concentration of particle per gram of tissue. And after a year, you can see on the right side, so there is a size difference between a small or a large nanoparticle, but we have about 2 milligrams of polystyrene accumulated per gram of tissue. So if you remember what I was saying, that people were alarming because they were performing ecotoxicology tests at very high concentration. Maybe they are not so far away from the truth. But there is discrepancy in the field because people see effect or not effect, and they don't understand why there is this discrepancy. And actually, there is a paradigm here that ecotoxicology tests report threshold effect concentration, which is the concentration at which we see a biological response, are reported using the concentration into the medium. So meaning that if you spike your water, you will use the concentration that you put into your water. But actually, depending the behavior of your contaminant, they might be absorbed or might be not. And actually, that's why you might or might not see a biological response. So I wanted to have a paradigm shift here with my work, because as I was the first to be able to quantify this concentration into the tissue, I want to push forward this aspect and change the way that we are reporting threshold effect concentration. So as I said, I work with 15 microgram per liter, which is the upper limit of the environmental concentration that we expect. So which means that you need to have a much more powerful or much more capability to detect the full range of environmentally realistic concentration. So that's why when I met Lucas and Daniele, I was quite excited about what they did develop. So they were developing a new interface on the Micadas for so what we call ADME experiment for biomedical application. And actually, this is the same kind of things that I'm doing, but for animals rather than humans. So I hope you went to Daniele's presentation on Tuesday, where he was presenting what he developed during his PhD, the double trap interface. This is quite interesting because as you do not have to pass by the graphatization for the sample preparation, you can have much more sample analyzed per day, and the hope for them was to go about 200 or even above this number of samples. And this is quite interesting in terms of ecotoxicological analysis, because when you perform an exposure experiment, you have a lot of sample, a minimum of 4 or 1,000 samples per experiment, and you need to be able to analyze them with a very good reliability and very quickly. So that's what we went for, using this double trap interface on the AMS. So the tricky part was to perform a very low concentration experiment, because when you are in your lab exposing those animals, you do not have an AMS next to you to measure, how much you are sparking into your water, and you have to rely on having a very good liquid scintillation counter. But we succeed to expose mussels at 140 becquerel per liter, which is about 200 ppt, and this has never been done before. So we did expose for 2 days to make sure that we will be having enough data point for the uptake phase, and the depuration was performed for 2 weeks. And the control we used was just mussels without any particles, and we used 2 different sizes to see the size effect about the exposure. So the tricky part, because I talked about that 2 years ago in Zurich at the conference, and we were struggling a lot about the sample preparation for the AMS, and the data was not so good, but actually we nailed it this time. So what we did was to extract the soft tissue of a mussel, freeze dry them, and instead of preparing a slurry or to grinding by hand, we used a mechanical grinder, and gave us a very nice sample preparation, because if you see, I can have an arrow somewhere, but I will just, yes. So you can see the sample here of a typical mussel. You have those black bits, which is more the stomach of, I would call that the stomach, which a biologist will kill me if I was saying that, but the stomach of a mussel. The white part is more the gills, softer tissue. So within the same body, the hardness of different parts of the body is not the same. And that's why we had a lot of problems previously, because a slurry will not be enough homogeneous to have a reliable measurement on the instrument. And the other aspect is that, and I'm sure you know that, when you prepare your sample for an AMS, you have to prepare about 100 microgram of sample, when the mussel is about 2 grams of sample. So you really need to make sure that what you're measuring is representative of the whole mussel. So this is why we went for a repeatability test, where we took 2 different sampling points for the uptake phase and 2 sampling points for the depuration phase, and we were very happy about the result because after 10 repeats for each point, at the uptake we have a variation about, for the highest 7%, and for the depuration we had the highest at 13%. But at those levels, this variation is really, really good. So we were very happy, and we could proceed with the full set of samples. So that's the result of this uptake and depuration. So in pink, you have the 20 nanometer, and in blue, you have the 250 nanometer. The equation you see there, do not be worried about it, and do not try to understand them, but basically this is the basic or standard way of looking at the bioaccumulation modeling. And what you can see is that the curve drawn for each fitting are good, but they are not perfect. And I will just talk about that next slide. But the most important thing here is you can see that the blue and the pink point are really different. So we have an uptake that is significantly different than much higher for the 20 nanometer compared to the 250. And you can see that the depuration as well, there is a significant difference between the depuration speed for those 2 size of particle, which means that there is still a size effect on this behavior of particle within the body. So as I said, the modeling that I used first was not perfect. So what I did was to look at, because the purpose of all this work is to be able to translate that into policy. And a policy needs to have a framework to work with and make sure that all experiments that you perform are comparable. There is a very nice team in Lyon in France where they developed this, what they call mosaic bioaccumulation modeling, which is a toxicokinetic modeling and respecting the OECD framework for policymaker. And what you can, so the first thing they do is a posterior predictive check, so meaning that do the model fit very well with your data point. So for the 20 nanometer here, you can see that the fitting is 98%, which is very good, meaning that there is only 1 point outside of this modeling. But what I would like to stress for this slide now is the time that we need to eliminate 95% of those particles, meaning that if you were in a perfect situation that there is clean water, you will need 113 days for eliminating 20 nanometer nanoparticles. And if you compare that with the 250, so the 250 was much better, the fitting was 100%, so less scattering, showing that there is a clear difference of behavior in the accumulation of those particles with the size. And what I want to stress is the time that you need to eliminate those larger particles, which is only 35 days. So you need 3 times less time to eliminate larger particles compared to the smallest particle, which means that we really need to understand what's happening with the smallest particle because they will be longer in your body. So as I said, those modeling is for perfect situation. And if you compare those different parameters that have been able to characterize with this modeling is that, and they are significantly different. So Ku is for the speed of uptaking those particles, and the speed for uptaking 20 nanometer is twice the speed of the 250. And for the elimination, the speed for eliminating those particles is 3 times slower for the small particles compared to the large one, which is in agreement with how much time we need to eliminate them. So what will happen in a real world? Because we can't really put them in a clean tank. And I'm still working on it because you saw the prediction of the 15 microgram per liter exposure. I'm still working on it to have this prediction, but what I can tell you is that as we need this time for eliminating those particles, we can expect to have an accumulation of those particles even at such low concentration. And I would say that we are exposed to those plastics since more than 60 years. So that would be very interesting to see the prediction over 60 years, actually. So what is the impact of that research? So we pioneered the use of the double trap interface AMS for ecotoxicological investigation, which will pave the way of a lot of work regarding that, because as the carbon-14 is already used for policy makers to inform those policy as a gold standard, but at much higher concentration, here with the capability of the CAMS, we will be able to have a real risk assessment of those particles, and we will be the first doing that reliably because we are labeling the carbon backbone of those polymers. But also, what is really interesting that you can look at metabolic pathway if you look at degradation of those plastic. Because if you have the incorporation of carbon in any metabolite, you can follow that, and the AMS will be probably the tool for doing that. And lately, I've been approached by some people in Netherlands to perform human health investigation and to be able to actually look into human health for this uptake and depuration of those particles. So that's a really exciting piece of work that I propose for an ERC starting rank, like Daniele talked about it on Tuesday, and I've been I would say, I couldn't believe that I've been awarded the starting grant ERC, but we will develop further this AMS approach for spatially resolved carbon-14 analysis, which is really exciting to look at laser ablation coupling for that. Yeah, so I'm open to questions now.
Okay, thank you very much, Maya. If you heard Maya, she's open for questions. Oh, we have questions.
Yeah, good. Do you want to?
Do we have? We have a help. Okay. So Walter, and then also. Okay. Oh, we have 3 questions. Okay. Ah, sorry.
Thank you very much for this enlightening talk. I just would like to understand the different sources of the plastics. We understand macroplastics, bottles. Microplastic already is more difficult to understand where they come from, and nanoplastic, I don't know. But is it actually a process that the larger species eventually end up as nanoparticles? So the origin of the original origin of large plastic things, or is this a wrong assumption?
No, that's correct. So actually, since 20 years, there's a lot of people fighting for banning microbeads or those kind of things. So now, a lot of West countries do ban them in products. So the main source is the degradation of your first piece of plastic. Okay. And, as there's weathering since more than 60 years now, they are fragmentating, and they are going smaller and smaller.
And so the time scale of that degradation is in the order of several years, or no?
Yes, several years, yeah.
Okay.
There is people collecting plastic on beaches, and I think 2 months ago there was someone collecting a bottle, and somehow on the bottle it was written 1989. So it was a bottle in the sea since 1989 or about it.
And the degradation process is understood?
It was degraded. You could recognize that was a bottle, but still a part of it was degraded.
Okay.
Thank you. The next over there.
I just want to make like a comment. So the talk is very interesting, and the motivation is very good, but the last few slides, the data you showed doesn't seem to support some of the conclusions. Because if you look at the data, the scatter is just too much, and worry about you may overinterpret the data a little bit. So in my opinion, you should introduce like something physicists use very often. It's called a goodness of the fit. It's called chi-squared.
Yeah, we have it.
Yeah, but we can look at those slides, and I can draw any curves like a straight line. It won't be different. You cannot tell the difference between those models. Some of the slides.
Yeah, on the slide, but I mean, I have the full.
You can go to one of those slides, like if you will, then.
That? Oh, sorry.
So for example, this one, I can draw a straight line, and the fit is as good as orange one.
Yeah, so this is always a difficulty with biological variation, and this is introduced into the framework of this modeling.
Right. So those parameters you can extract, right? But then you should give uncertainties to those parameters. Then because you compare the parameters for different sizes, 20 nanometer and 250 nanometer, and you said there's some difference between those parameters. But if you take the uncertainties into account, then there really is no difference between them then.
Well, but I mean, there's fitness.
Goodness of the
goodness fit. Of the fit provided, and so this kind of thing has been tested.
Right. You understand what I'm saying, right? So for example, this data, you have a curve, right, like this, but if I just draw a straight line, a constant.
I'm very sorry, but I really think that we spent a lot of time. Maybe just one more question, but very short, please. I'm sorry. Maybe you can continue email. Yeah. Only one question.
Another question. Since mussels are filter feeders, you showed very nice accumulation rates. Do you have any ideas about other marine organisms? Like, does nanoparticle just flow through the gills of fish, for example, and not really accumulate? And if the accumulation rate is an order of magnitude smaller, are you still able to detect and follow metabolic pathways?
So I did test on fish, and the result is actually they are translocated, so they are going through all the membranes, and they are even reaching the bones. Yeah, that was not the presentation today, but that's the result. So the hypothesis is that we might have a transport through blood cells, because they might have the capability to be translocated into cells and be transported with blood cells. I didn't do yet any metabolic pathway, but that's the hope that with the power of the AMS we will be able to detect this kind of incorporation, because the plastic is not 100% degraded, because you can't know. If you look at total degradation, it should be CO2, and a lot of results show that maybe less than 1% is transformed as a CO2, but you might have several products of degradation, and we will be able to track them if it's the case by labeling them.
Okay, I'm sorry, I think we don't have any more time. Maybe by email or something next time. Okay, so thank you very much once more. And now we approach our final lecture for today and for this section, and also the entire conference. And the presenter of the lecture will be Henrik Grotter. with the presentation, high-resolution mapping of DIC, DOC, and POC radiocarbon distribution in the northwestern African upwelling system of Mauritania. So, please.
Almost. So apparently you can see I changed the title.
Oh.
And it's a pleasure giving the last presentation of this really wonderful conference. And today I'm going to talk to you about how we can use radiocarbon to understand the partitioning of the 3 marine carbon pools in the ocean, and how we can draw information or decouple information to understand local oceanographic features. And most of my talk will be focused on the marine carbon cycle. And in the ocean, we have 3 carbon phases. Mostly, and the biggest one is dissolved inorganic carbon. And we have 2 organic carbon fractions, namely dissolved organic and particulate organic carbon, which can be separated by filtration. And really everything I'm talking today about is a biologic carbon pump. And essentially, CO2 is taken up in the ocean by gas exchange and is forming the DIC pool, which is then used by phytoplankton for photosynthesis, which is essentially fixing the inorganic carbon, forming particulate organic carbon. And the particulate fraction is intensely recycled in the water column by microbes, other microbes, and can be oxidized back to dissolved inorganic carbon, or basically broken down into small molecules, which is then forming up the dissolved organic carbon fraction. And this particulate fraction is actually sinking throughout the water column. It has sinking velocities of a couple of 100 meters per day. And during sinking, the POC is consistently reworked by microbes. And essentially, only about 5% of what was formed, in particular form in the ocean surface, will actually end up on the seafloor. And this process is called biologic carbon pump, and the efficiency of the biologic carbon pump essentially defines the sequestration potential of an ocean. And when we now add our radiocarbon information to this, we can draw very basic assumptions of how radiocarbon should behave for the individual carbon phases. And this is really inspired by the excellent work of Alan Druffel and their colleagues at UCI. And for dissolved inorganic carbon, we know that it is in equilibrium in the surface water, and during sinking, it will decline based on the water mass distribution. Particulate organic carbon, considering that it's formed in the surface layer and has sinking velocities of multiple 100 meters a day, we don't really expect any depletion on the 14C for the particulate fraction, and dissolved organic carbon is a bit of a beast, and depending on the fraction of the DOC you're looking at, it will behave differently, but very basically it is younger in the surface and it will be very depleted in the deep ocean. So in the deep ocean we can reach data 14C values of minus 500 per mil. And this assumption is pretty much true if we go to an open ocean situation. But what we are interested in are local phenomena that occur close to the shore, so along the shelf and on the shelf break, because we want to understand how carbon is transported in the ocean and how it is affecting the efficiency of the carbon pump. And when we are in the coastal regions, we have primary production going all the way in the top layer, and it's then eventually sinking. But what else we have is a massive amount of transported material, organic or inorganic, coming from the shelf, and then you have resuspension of sediments on the shelf breaks, and et cetera, and these resuspension material will be transported throughout the water column. And the assumption is that we can detect so we call that nepheloid layers. And the idea is that we can detect the nepheloid layers and the processes that lead to them by deviation from our expected radiocarbon trends. And to test this hypothesis, we have the great opportunity to go to Mauritania, so that is the Atlantic Ocean, and we're in Western Africa. And we go to that area about every 2 years, because there's a long-term monitoring station. And we have a lot of information about the sedimentation processes in that area. And basically what we do is we collect very high resolution, spatially high resolution sample sets. Shown here are two parallel transects from the shelf towards the open ocean. And today I'm going to talk about only the northern transect, but the southern is basically showing the same thing. These samples were collected in 2020, and this area is wonderful because it has year-long upwelling, and we know for sure that we have sedimentary suspension going on along the shelf break. Further, this area is quite simple because we only have to deal with marine organic matter, because there's essentially no terrestrial influence from Western Africa. There are no rivers, there's almost no vegetation. So everything we deal with is purely marine. In the field, we do perform our high-resolution sampling using CTD rosette systems, which is lower to a certain water depth, and we collect water. Which gives us the dissolved inorganic carbon fraction. And depending on the carbon or expected carbon concentration, we will filter 5 to 70 liter on GFF filters, which gives us the particulate fraction. And it's important, I'll call this material coming from the CTD as particulate organic carbon suspended. The reason for that comes later. And whatever does not stick on the filter gives us the DOC fraction. The high resolution sampling is paralleled with low resolution sampling using in situ pumps, which are also lower to similar depth, and there we can filter up to 900 liter per deployment. And that fraction we will call the particulate organic carbon sinking fraction. And you can tell by the number of samples that we collect, we need relatively high throughput of measurements, so we decided to use our Micadas AMS and have analyzed all samples as CO2 gas using the GIS interface. Important to note is that we are not reporting bulk dissolved organic carbon radiocarbon values, but only the solid phase extracted material. That brings me straight to the data. What you see here are the 4 stations, and going from right to left, the gray areas indicate the depth of the seafloor. So on the right side we are on the shelf, and the left side would represent the open ocean. On the y-axis you have the water depth, and on the x-axis you have the measured data 14C value, and the blue line indicates the data 14C of the dissolved inorganic carbon, which really shows what we would expect. On the shelf we have primarily atmospheric signal, and the further we go offshore and in deeper waters, the DIC is aging. And this is pretty much also true for the solid phase extracted dissolved organic carbon, which is also declining with water depth, but is significantly depleted relative to the dissolved inorganic carbon. So DIC and DOC are really behaving as we would expect based on our hypothesis. It's different when we look at the particulate, so the suspended particulate organic carbon fraction. This is really, it's nice, it's showing a decline with depth. But if you remember what I told initially, we would have expected that due to the high sinking velocities, we would not see any apparent aging in that fraction. So there's clearly something going on here, and the suspended fraction is significantly depleted relative to what we would expect. And there are now small tiny triangles that have appeared. You can see that on the very far left. And these triangles show the 14C value of the sinking fraction, so the large volume sample. And we see that it is in agreement with what we would expect. We have basically a sinking signal. But we see this clear difference between the suspended and the sinking particulate organic carbon fraction. And in the ocean, the radiocarbon distribution is mostly controlled by the ocean currents, so everything should be in the first order controlled by water mass movements. And if we analyze our data based on the water masses, which are now shown on the left, and I'm not confusing you with all the names, we can simplify that and group all the water masses into 4 major water bodies. On top we have the surface water, central water, intermediate water, and deep water. And the water masses are now separated by vertical lines. And if we now plot the radiocarbon value of the individual samples that come from these specific water masses, we see clear, statistically robust separation between the water masses for DIC and also for dissolved organic carbon. So both phases are controlled in the first order based on the water mass distribution and water mass movements. And yeah, the POC doesn't. We can tell, okay, the surface layer is a bit different, but basically below the surface layer, we don't see any water mass dependency. And the data really, if you look at it, is wild and varied, and that's a good thing, because that's actually what we're after. Because if we know that the suspended fraction is not controlled primarily by the water mass movements, we can actually try to find local phenomena and local processes that are hidden underneath the data. And what we're really after is the spatial distribution, because we want to understand, do we have actually resuspension, and how is that influencing further offshore? And when we just look at the DIC and the DOC fraction, we don't really see any patterns occurring because it's controlled, as I said, by the water mass movement, and it's basically just decaying, or not decaying, declining with water depth. It's different when we look at the particulate suspended fraction. We can identify 3 major areas in the ocean on that particular site. The surface layer is relatively modern, going down to -100 per mil. Then we have between around 1,000 meters, we have this very depleted values in a suspended fraction around -300 per mil. And then underneath, we have a layer where the data potency ranges about -200 per mil. And we can conclude that the top part represents primary production signal and shelf export, whereas the bottom part, we suspect that it reflects sedimentary suspension, because the analyzed potency of the suspended fraction actually mimics the isotopic composition of the surrounding sediment. And then we have this blob in the center where we really don't know what's going on there. So currently we don't have a proper explanation, but we can maybe draw some conclusions from it, what we see. So what I've calculated here is the delta 14C, so the isotopic difference between the sinking material and the suspended material that we analyzed. And you can see that with the red color, we basically have no difference, and the blue color shows we have a large isotopic difference between sinking and suspended. And again, obviously, we have this center water mass where we have a very depleted suspended fraction, and we conclude kind of that this represents something like a buoyant POC phase, which is not sinking, but due to the fact that it has similar densities as the surrounding water, it will move vertically rather than it would move laterally instead of vertically. So it's not sinking, it's just sitting there. But what can that be? And because we have analyzed all carbon phases, we can use a 2N member mixing model between the particulate organic carbon from the surface and the DOC radiocarbon value in a specific depth. And if we calculate that, I'll call that fraction DOC, if we calculate that, we see that in the same area as we have this very depleted suspended fraction, we find that we have the highest abundance of dissolved organic carbon radiocarbon signal in my suspended fraction. And up to 95% actually. And we have been thinking about this for a while. Actually, this is the 3rd time I'm generating that kind of data set. And we have modified our sampling approaches 3 times, and we're still getting to the same patterns. So we're starting to believe that this is actually true. So if it's not a sampling bias, what could it be? So there's a hypothesis that dissolved organic carbon molecules can find each other and attach to each other and essentially form microgels, which are then characterized technically as particulate organic carbon, because these microgels form millimeter-sized aggregates which would sit on your filter, but technically they are dissolved organic carbon fractions. So they are buoyant, they are not sinking, they are just forming in the water column. And what does it actually mean? So if we want to use the POC flux to estimate sequestration rates and the efficiency of the biologic carbon pump, what we're observing here is potentially leading to a significant contamination of that data based on this in situ production of POC, which has no influence on the flux rates of particulate matter. So with that, thank you.
Thank you very much for a nice presentation. We have a time for 2 or 3 questions, so if there are some questions. Okay, we have a question.
Thank you. And I was wondering, did you do the measurements at like specific set intervals, or did you take more measurements in the surface, or closer? Did you have a closer distance between the measurements in the surface layers of the ocean?
There are 2 hypotheses. So basically, you can sample features, or you try to sample equal distance. This is mostly equal distance. But this equal distance is different because you have in a surface ocean, you only need 1 CTD bottle to get enough carbon, but in greater depth you need to collect 12 of them, basically. So it is depending on the carbon concentration at a certain depth. So we have a relatively high density in sampling resolution in the upper layer, but the deeper we go, the coarser the resolution becomes.
Have you tried to relate this to how the top layers of the ocean will be more mixed than the bottom layers of the ocean?
Yeah, the top layer is well mixed, yeah.
Okay, do we have more questions? Okay.
Thanks for the talk. I have 2 questions, but one very similar to what the lady asked. Okay. Is it your POC is not including black carbon, is that right?
Based on the solar phase extraction, I would think it is absorbed. It includes some sort of black carbon, yeah.
Okay, that's still included black carbon in the POC.
Chemically it should, yeah.
Okay, that means the black carbon will take a long time to, or change, right?
Yeah, right. So the DOC radiocarbon distribution is well known. So that is, whatever you measure in terms of DOC radiocarbon is always a mixture of the labile fraction of DOC, which is formed in the upper ocean, and the relative mixture to the refractory fraction of the DOC, which contains black carbon and all sorts of molecules that are simply not degrading with time. So that's why you have radiocarbon ages of, well, a couple of thousand of years in deeper oceans. So the material is to, and yet not completely resolved process accumulating in depth.
Okay.
Okay, if we have some more questions. It seems to me that is... Oh, yes, we have. Okay.
Kind of expected that.
Yeah, the last minute.
I'm curious about the DIC, actually. Look like the measurements was not done during the upwelling period. And how long is the upwelling?
The upwelling, so the main season of upwelling is in January, February, and the samples were collected in August. Next year, I'm doing peak upwelling to do the same sampling approach and to see how it differs due to upwelling.
Cool. Thank you.
Welcome.
Okay, so it seems to me that we don't have any questions anymore, so let's have a chance to applaud once more to the... And also to all other presenters and to organizers, so the organizers now have something to say. Bye.
Okay, so I don't get introduction, so I I have the honor, I think, of the conference summary and instructions for publication. And don't go after that because I heard there's some awards. And I don't know what else because Lucio always has something extra that you don't know yet. So, let's see if I can make this thing work. First of all, I think we should congratulate the Salento team. We can do it also now and then again at the end of my talk. So. Because we have a great trip to Gallipoli and a field trip to the lab and yeah, the fantastic party. So I don't know, in future Radiocarbon, I don't know what they have to do to top this. But maybe somebody will find something. So we also do some science and okay, my summary is a bit biased to the beginning of the week because by the end of the week I hadn't, you know, there was a big party yesterday. So anyway, there are many great presentations, especially by some early career scientists. I think there's some award coming at the end. So, you know, all kind of sediment preparation techniques, which is always a messy business in radiocarbon, methane sources. I really like the pollen separation method by a Japanese colleague, and also all kinds of other studies, especially today is not on my slide because I made this slide yesterday about the marine studies, and the microplastics is a fantastic talk. And so I don't really need to go through this list. I think the new techniques are really important, especially the laser-based saturated cavity ring-down spectroscopy has a lot of potential in the future for radiocarbon. Of course, also atom traps, maybe for other nuclides, and C14 in the atmosphere and terrestrial environment had a lot of good talks. And also some people sang songs. Today nobody sang a song yet. Anyway, maybe later. And so these are some other things I learned. You shouldn't drink cappuccino after 11:30. Okay, I asked many Italians. Nobody ever explained to me exactly why you shouldn't drink. Because, okay, in America you can buy cappuccino anytime, you know. So you savor the local cuisine, which is fantastic, and we all had many discussions about over wine, coffee, Aperol. And I hope everybody learned something new. Also, we learned to sing at the end of the talk. This is a new introduction, I think, for the future radiocarbon. And so then I have some advice, which is everybody can sing volare, oh, but the second question is the vincero is very difficult, and obviously I can't do it. But if you can do this, you shouldn't stay in science. Because of more money, you know, Luciano Pavarotti, he made a lot of money. Anyway, and also we had a great party, and what else do I have here? Also now I have some serious things about the publication. So this is the conference publication, which means these are the rules, about 4,500 to 5,000 words, 4 figures, 2 tables, supplementary online materials. This means about 8 printed pages. We're kind of flexible about this, but not too flexible. So if you send like a 20-page paper, that's probably not reasonable. There is no page charges for these proceedings. Obviously, if you have open access agreement, that's good. What else? Okay, there is no page charge. So if you have something with a lot of color figures, and the color figures are, you want them online in print, this costs extra. Online color is free, in print color costs money. So if you don't want the print copy to have color, you should think that make sure that your figures look good in black and white as well as in color. And if you really do want to have the color print, then there's extra charge. If for some reason you can't pay, you can talk to me or Kim. And there are some future conferences. I think the first one is next summer in Krakow in Poland for the Radiocarbon conference, which doesn't have an official date, but I heard some rumors that these are the official dates from IRCA and some Polish groups, beginning of July next year. And in 2026, there will be a radiocarbon archaeology conference in Niterói. And then there is C14, oh, there are 3 conferences, Clara 3 and C14 and Diet, which is somehow combined between a Brazilian group and a group in Chile. And I think we'll learn more about that later, unless anybody wants to say anything about it now. No? No? Say anything? No? End of June or beginning of July. Right, so these meetings will be somehow coordinated, let's put it that way. And let's see what else. Okay, thank the organizers again, but we can do that at the end because we're going to sing, right? And if you have any problem about the guidelines for the submission of the paper, contact Kim. She's always there to help. And it's too bad she didn't come to this meeting. Maybe she goes to the Radiocarbon meeting next year. And finally, we're going to sing in a minute. I take this picture because Wei Jiang should send me this picture, so I borrow it, but I put the copyright at the bottom, so it's okay. So now, Volare. Let's go again. I don't know that word. That's some announcement from LECCE.
So I think you probably miss your families and friends, but I'm almost sure next week you will miss this community as well. So to give you some hope, I have some short advertisement. Oh, sorry, this one. Yep. About the next conference, which is approaching very rapidly. We have to wait a minute, a second. Okay. So the next conference will be in Krakow, which, which is over there, and there is a strict connection because in Krakow, the most distinguished queen of Poland was named Bona Sforza, and she came just nearby from Brindisi, so to Krakow. So we have a very strong connection, I would say, and so you will be like home when you go from here to Krakow. Yeah. So sorry, I have to go back. Where's the page back? The conference will start on end of June and will finish about 5th of July, probably. You can find the, maybe not because of the plant, the QR code to go directly to the conference website. The website is more or less running, but with some very basic information, which I will give you as well right now. So. It's easy to get to Krakow because we have a quite large airport nearby, and it's like 25 minutes, depends on the traffic, ride to the city center from the airport. There are very convenient trains going from Kiev, Prague, Bratislava, Berlin, Vienna, Budapest, and Warsaw as well. So you can get all of these trains. There is a A4 highway going from Germany through Krakow to the eastern part of Poland. Coaches, very cheap way to travel from all around the Europe, and boats. You can theoretically go by boat from Baltic Sea to Krakow, but we do not recommend it definitely. The University of AGH University of Krakow is quite old university. I mean, not like the one in Edinburgh, for example, which was mentioned a couple of minutes ago. But we have a large venue, very, let's say, almost in the middle of the city. So everywhere is just within walking distance. And yeah, so you may expect usual topics we cover during Radiocarbon conferences, but if you are interested in something new, let's give us some information about it, and we will try to make it happen. So as you can see, the venue is over there, and plenty of different places you may go and enjoy, not only the conference talks, but also the city, like here. Very similar city to this, except the Krakow is much larger than Lecce. And yeah, so the leisure time. We plan some pre or post-conference tours. For sure, we don't know which one will be available and if you will be able to join all of them. But main places you may try to check right now is Wieliczka salt mine, which is a very nice place to go. Wawel Royal Castle, because Krakow was a royal city for many hundreds of years. And so you may visit Royal Castle as well. Jewish quarter, Kazimierz, which is part of Krakow, and some who is very brave enough to see this, Auschwitz. So here is the way to find us. Radiocarbon 25.online is the easiest way because it's easy to remember, and this is just the redirection for the Radiocarbon 25.agh.edu.pl. And you can also try to monitor us on X or former Twitter. We'll try to update information there as well. So thank you very much, and let's hope to see you all next year in Krakow. Thank you.
So before concluding the conference, you remember we decided to have 2 awards for the best talk and for the best poster for young scientists. So team, please come and say yes. Who are the winners?
So I have to say it was a difficult choice because there are many good early career scientist presentations, and so we evaluated the presentations on quality and clarity and confidence. And you know, there's also a question of confidence, especially for people who the English is not their first language. So we decided that we have a small committee. Okay, so we met, and we believe that Sophia Bjornheim should get the award. So welcome, Sophia. And also there's an award for best poster. And so we evaluate the poster based on, the style and not too many words on the poster. and Lucas Ngoyen is the awardee. I hope he's still here. No? No? Oh, he's there. Okay. So I heard there's some other awards, but
yes. The most important award of the conference, if you remember. This was the proposal, the Golden Google Award for the best singer. Who didn't sing this song was eliminated, as I told you before. And the second proposal was this. Three finalists. The first one is Monty Ochoki. Please come. I didn't take the great responsibility to choose the winner. So there are three finalists. The first one is Monty Ochoki. And the community has to vote. Please, you have to repeat your performance.
Volare, oh, cantare, oh.
And the second finalist is Natalia Piotrowska.
Volare, oh, cantare, oh,
The third finalist is Timo Reiner.
He's almost Italian, he did like this.
Vincerò, vincerò.
So now you have 1 minute to vote.
We have to count the ballots? No?
But we have another special award. Artificial Intelligence Golden Google Award. Do you remember? Alexander Cherkinsky said, "Thanks, sorry, but I don't want to compete with him." But we can do a lot to help him. Okay. I wait now for the committee, please. Let's give us the winner, the real winner.
Yes, yes.
Sorry? No, they will have something different.
So you know, we check that you know this vote counting is all ethical, you know, above board, you know, as far as we know.
Unfortunately, Alexander is not here. He has left, yes. So we will send him the award. Suspense. And the winner is Timo. Okay. We are just at the end. Let me thank all the supporters, all the sponsors of this conference, the University of Salento, all the others, and of course the people, the Applied Physics group, starting from Professor Dr. Gianluca Quarta. He did a great job. I will call them. Giorgio Carbone, please. Marisa, Lucio Maruccio, Vito, Mattia, Dalila, Sabrina, Stefania, Ritz, and the staff of the organization Modus Animi, Marta, Elisa, Alice, others are not here for academic reasons. Many thanks to all of you. And we hope to see you again in Lecce or in the near future somewhere else. Ciao, bye-bye.
And at the last but not the least, a great thanks to Professor Lucio Calcagnile.